Welcome to The Explainer. Today, we are diving headfirst into a future that might sound like science fiction, but is rapidly becoming science fact. We're talking about a world where the very lines between biology, technology, and design just completely dissolve.
So let's kick things off with this really radical idea from our source material. What if our bodies aren't some fixed piece of hardware we're stuck with? What if, instead, they're a programmable platform? You know, like an operating system that we can finally access.
And that really is the question at the heart of this entire explainer. We're going to unpack a set of technologies that could take us from just healing the body to literally writing brand new architectures into it. Okay, let's get into it.
To even get our heads around this, we have to start by completely changing how we think about disease. For centuries, we've had one very specific way of looking at illness, but what if that whole framework, that whole metaphor, is just wrong?
Think about it. We always use the language of war. We fight cancer. We're attacked by viruses. But what if disease isn't an enemy? What if it's just a bug in the software? Cancer is a corrupted loop in a cell's program. A genetic disorder? It's just a typo in the source code.
And if disease is a bug, then the cure isn't a weapon, it's a software patch. I mean, that one shift in thinking changes absolutely everything about medicine. So what's the tool? What's the USB drive we use to install this patch into our biological computer?
Well, this is where it gets really interesting, because we're going to need a surprising ally. Turns out, the very thing we've always thought of as the enemy in that old war metaphor, it might just be the hero in this new one.
So this is the crucial shift, right? We're not fighting an invader, we're correcting an error. And viruses, which have literally spent billions of years perfecting the art of getting inside our cells, well, they become the absolute perfect delivery vehicle for our little software patch.
So here's how it works. Scientists can take a virus, something like an AAV, and they basically gut it. They strip out all the harmful code and replace it with new constructive instructions. And this is not sci-fi. FDA-approved therapies are already out there doing this. A therapy called Luxturna uses one of these viral nanites to deliver a correct copy of a gene into the eye, literally restoring vision to people. It's amazing.
And if you want to know just how effective this can be, look at this number, 93.5%. That is the success rate for Casgevy, a therapy that uses CRISPR. You know, the molecular scissors that can cut and paste DNA? It effectively cures sickle cell disease. In the trials, 29 out of 31 patients were totally freed from the awful pain crises that come with it. It just goes in and patches the broken code, right at the source.
Okay, so we have a delivery system for our code. That's a huge deal. But what if we could use a similar kind of biological logic not just to heal, but to build? This is where the story takes a really fascinating turn, by looking at another biological agent that's famous for all the wrong reasons. Let's talk about prions. Yeah, I know what you're thinking. Mad cow disease. And you're right, they're known for causing these devastating neurodegenerative diseases. But the very thing that makes them so dangerous, their ability to misfold and basically convince other proteins to do the same, is built on an unbelievably stable and strong structure. It's called the cross-beta architecture, and it makes them one of the most incredible materials ever discovered.
And this table just shows you how wild their potential is. You can see right here that engineered amyloid fibers, that's materials based on this prion logic, they don't just match, they actually surpass the toughness of spider silk. And we all know spider silk is the gold standard for biological fibers. Now, comparing it to steel is tricky because steel is about 8 times denser, but trust me, on a weight-for-weight basis, these fibers are phenomenally strong. This isn't just a material, it's a construction kit given to us by biology.
All right, so we've got our delivery system and we've got this amazing building material. But how in the world do you control it? How do you tell these materials what to build and, just as importantly, where to build it? Well, the answer is surprisingly elegant.
So it all comes together in this 3-part system. It's beautiful. First, you've got the viral nanite, which delivers the instructions. Second, those instructions tell the body how to make the self-assembling prion fibers. And the third piece? The magic ingredient, the control switch that tells them exactly when and where to start building, is just light.
This whole process is a form of optogenetics, which is really just a fancy way of saying we're using light to control cells. By fusing the prion-forming protein to another protein that's sensitive to light, a simple beam of light becomes a trigger. It tells the fibers precisely where to start assembling, with microscopic accuracy. We are literally using light to print solid structures out of a liquid. It's programmable matter.
So what do you do once you can actually program matter itself? Well, the applications are just staggering.
We're moving way beyond medicine now, and into material science, computing, you name it.
Okay, just imagine this for a second. A spaceship hull or a deep-sea submarine that can fix its own cracks. If you make the hull out of these fibers, any damage would expose the ends of those fibers, which would then automatically trigger them to start regrowing and weaving themselves back together, sealing the breach. It's a living, self-healing material.
Or how about this? We could move beyond silicon and start building computers at the molecular scale. In this vision, DNA could store unbelievable amounts of data, while these prion fibers would act as the self-assembling logic gates and wires. We'd be creating processors with an information density that would just dwarf anything we have today, all built from the bottom up, just like life itself.
And this one might sound like pure sci-fi, but this technology could lead to something a lot like the food replicators from Star Trek. Seriously. By using light to assemble protein lattices from a liquid soup of ingredients, you could perfectly mimic the texture of meat or fish or anything you want. You could create food on demand anywhere, just by changing the light pattern.
So this all sounds incredible, right? Game-changing. But this kind of power, it brings us to maybe the most important question of all. Just because we can write new code into the book of life, should we? And who gets to hold the pen?
The first, and maybe most obvious challenge, is access. I mean, look at this. With therapies like Casgevy already costing over $2 million per treatment, we're staring down the barrel of a biological class divide, a world of the enhanced and the unenhanced. And that's a split that would be far more profound than any economic inequality we know today.
And the questions, they get even deeper. How do you get consent for a change that doesn't just affect one person but could be passed down for all time? Who gets to make those kinds of permanent, heritable edits? Should we build in kill switches to reverse these things if they go wrong? It forces us to think really hard about governance and how on earth we prevent a biological arms race before it even gets started. And that brings us to this final, really provocative thought from the source material. For all of human history, we have been subject to the whims of our biology. For the very first time, we are learning to pick up the quill and write the code ourselves. The question isn't if this is coming. The only question is, what will we choose to write?
What do a virus, a deadly misfolded brain protein, a beam of light, and like a floating weed in a Nebraska ditch have in common?
I mean, it sounds like a setup to a really bizarre joke.
It really does. But today we are going to look at how these four seemingly unrelated things are actively becoming the hardware, the software, and, well, the infrastructure of the next phase of human civilization.
Which is a massive claim, but the data is there.
Oh, absolutely. Welcome to the deep dive. You are here because you're someone who craves the big paradigm-shifting, aha moments, but you absolutely do not want to drown in a textbook to get them.
Right, no one has time for that.
Exactly. So our mission today is to take a shortcut to the future, unpacking a massive, unified architectural vision laid out in a stack of 4 heavily researched essays.
Yeah, these were written by independent researcher Zachary David Enfield back in March of 2026.
And the scope of Enfield's work here is it's staggering.
It really is. He titles this unified framework from the ground up. And I mean, the essays span everything from the absolute bleeding edge of molecular biology and quantum materials all the way out to civilizational infrastructure and large-scale agriculture.
It's so broad, but there's a central unifying thesis across all these disciplines, right?
Yes, there is. The core idea is that humanity is in the middle of a profound transition. We are shifting away from treating biology as this mysterious, fixed system that we just, have to endure.
Right, like it's just a lottery.
Exactly. And we are beginning to treat it as a programmable, customizable substrate.
A programmable substrate, meaning we're basically treating life itself like technology.
That's it, exactly.
And to really grasp how radical this shift is, I think we have to start with Enfield's first premise, which is the language beneath the flesh, like how we go about reprogramming the human body.
Yeah, and he points out that for our entire history, we've talked about disease using the vocabulary of war.
Oh, totally. Like you fight off a cold or you declare war on cancer.
Right, we rely heavily on militaristic metaphors. A virus is an attacker. Cancer is an enemy force invading the body.
But Enfield argues that this framework isn't just outdated, it's actually mechanically incorrect.
That's right, because beneath every symptom, from an autoimmune disorder to a tumor, there is a more precise truth. These conditions are not enemy combatants.
They're just, what, glitches?
Basically, yeah.
Yeah.
They are simply failures of information. They are corrupted code running in the living cell.
Mm-hmm.
And, you know, the living cell happens to be the most sophisticated, densely packed computational substrate that evolution has ever produced.
Wow. Okay, that reframing really changes everything. Because if a disease isn't an invading army, but rather just a coding error, then our traditional medical tools suddenly look, well, incredibly barbaric.
Yeah, they really do.
Like, if your laptop has a software glitch, you don't cut the hard drive out with a scalpel.
No, you definitely don't.
And you don't poison the entire motherboard hoping the glitch dies before the computer does, right? You just write a software patch to debug the code.
Exactly. And delivering that patch requires a very specific kind of USB drive, essentially.
A biological USB drive.
Right. Human engineers have spent decades trying to build microscopic delivery vehicles from scratch.
Mm-hmm.
Synthetic lipid bubbles and polymer shells, just trying to get therapeutic code inside our cells.
But Enfield points out a huge irony here.
He does. He says nature has already spent 3 billion years perfectly optimizing the ultimate nanoscale delivery vehicle. We already have viruses.
Okay, let's unpack this. Because the idea of using a virus as a medical tool feels so counterintuitive. I mean, we've spent our whole lives terrified of these FedEx trucks because they've always shown up at our cells delivering bombs.
That's a great way to put it. The FedEx truck analogy is biologically spot on.
But Enfield is saying, look, the truck itself is brilliant, right? It knows exactly how to navigate the highways of the bloodstream. So what if we just intercept the truck, empty out the explosive payload, and put a genetic repair kit in the box instead?
Yes, because a virus, at its core, is just an extraordinarily elegant container. It has this outer protein shell called a capsid.
Okay, caps.
And over billions of years, that capsid has evolved something called tropism. Tropism means the outer shell has specific chemical keys that only fit the locks on very specific types of human cells.
Ah, so like a respiratory virus has keys for lung tissue.
Exactly. And a hepatovirus has keys for liver cells. It knows exactly how to float through the chaotic environment of the human body, ignore billions of irrelevant cells, dock with its specific target, cross the cell membrane.
That evade our internal immune alarms.
Right. It does all that, and then it injects its contents directly into the cell's nucleus.
It's basically the ultimate precision-guided microscopic syringe. And the text notes that the scientific community is leaning heavily into a specific type of virus for this, right? The adeno-associated viruses, or AAVs.
Yes, AAVs are currently the gold standard platform for this kind of gene delivery.
Why them specifically?
Well, they are incredibly small, and they naturally do not cause any known human disease.
Which seems like a huge plus.
It's massive. It makes them incredibly safe starting points. Plus, they don't have a complex lipid envelope, which makes them physically stable.
Oh, wow. So how widely used are they right now?
Well, as of 2024, there were already 7 AAV-based gene therapy products fully approved by the FDA, with over 110 active human clinical trials ongoing.
That's insane. And the payload we are packing into these AAV delivery trucks is frequently CRISPR-Cas9 technology, right?
Yes, the famous molecular text editor.
I think most of us have, heard the acronym CRISPR, but help us visualize how this is actually debugging the code inside the cell.
Okay, so think of CRISPR-Cas9 as a 2-part biological machine. The first part is the guide RNA. You can actually program this piece of RNA in a lab to perfectly match the exact sequence of the corrupted DNA code you want to find.
So it acts like a molecular GPS coordinate.
Exactly. And the second part is the Cas9 protein, which is an enzyme that acts like a pair of microscopic scissors.
Okay, so the AAV truck drops this whole package into the cell, and then the guide RNA just floats through the massive library of your DNA until it finds the exact typo.
Yes, it finds the typo, binds to it, and then the Cas9 scissors make a precise targeted cut in the double helix.
And then what happens? Doesn't cutting the DNA damage it?
Well, once the DNA is cut, the cell naturally tries to repair it. We can harness that repair process to either permanently disable a faulty gene or, and this is even more incredible, we can provide a corrected template for the cell to use during the repair.
Wait, really? So literally overwriting the typo with healthy code?
Exactly that.
And the examples Enfield highlights to prove this isn't just, science fiction are staggering. He points to Casgevy. This was FDA approved back in December 2023. It uses CRISPR to edit a gene called BCL11A in a patient's own stem cells to cure sickle cell disease.
Yeah, sickle cell is a devastating genetic disorder. Just a single letter mutation in the DNA causes red blood cells to fold incorrectly into rigid sickle-like shapes.
And those malformed cells get stuck in blood vessels, right?
Yep.
Causing excruciating pain crises, organ damage, premature death.
Yes, it's horrific. For decades, it was managed with painkillers and blood transfusions. But Casgevy intercepts the corrupted instructions. By editing that specific BCL11A gene, it essentially forces the body to switch back to producing a fetal version of hemoglobin.
Which doesn't sickle.
Exactly. It completely bypasses the problem.
And the results in the trials were just unbelievable. Like 29 out of 31 patients were completely free of severe pain crises for at least a year after a single treatment.
It's life-changing.
It's a permanent patch. It doesn't just treat the symptoms, it fixes the corrupted instruction set.
The implications for chronic illness are profound. You eliminate the lifelong dependency on chemical management. Enfield actually highlights another therapy in the same vein, CDX310.
Oh, right, the cholesterol one.
Yeah. In a 2025 trial, researchers used a single infusion to deliver an edit that simply turns off the ANGPTL3 gene in the liver.
And what does that gene do exactly?
It regulates lipid metabolism. By turning it off, patients saw their LDL cholesterol drop by a staggering 50%.
From a single infusion.
A single infusion.
Yeah.
And you permanently rewrite the metabolic pathways that lead to heart disease, which, by the way, is the leading cause of death globally.
It's incredible. But Enfield doesn't stop at curing existing diseases. He takes this logic and drives it right into the realm of biological enhancement.
Yeah, this is where it gets really wild.
He introduces this concept of functional translation. Basically, the idea that evolution has already solved almost every physical extreme on Earth. Radiation, freezing temperatures, lack of oxygen.
Right, those survival mechanisms are just lines of code written in the DNA of other species. The biological diversity of this planet is essentially an open source code repository.
That's such a cool way to look at it, an open source code repository.
If an organism has evolved a unique mechanism to survive extreme stress, that mechanism is encoded in its genome. Synthetic biology gives us the tools to read that code, translate it into a format that human cells can understand, and then use our viral nanites to install it.
And the wildest example he pulls from the repository is the tardigrade, right? The microscopic water bear.
Oh yes, the legendary water bear.
These things are insane. You can freeze them, boil them, dehydrate them for a decade, or literally shoot them into the vacuum of space and they just wake right back up.
They're nearly indestructible.
And crucially, they can survive radiation doses thousands of times higher than what would turn a human into a puddle. Enfield explains this comes down to a specific protein they produce called D-soup.
Yeah, D-soup. It stands for damage suppressor. When researchers looked at the physical mechanism of how this works, they found something remarkable.
What did they find?
Inside the nucleus of a cell, your DNA isn't just floating loosely, right? It's tightly spooled around proteins to form structures called nucleosomes.
Okay, I'm picturing like thread on a spool.
Exactly. When ionizing radiation hits a cell, it splits water molecules, creating highly reactive hydroxyl radicals. These radicals tear through those nucleosomes and shatter the DNA strands.
Which is what causes radiation poisoning and secondary cancers.
Correct. But the tardigrade's D-soup protein actually physically binds to the nucleosomes. It drapes over the spooled DNA like a microscopic Kevlar blanket.
A microscopic Kevlar blanket, I love that.
It acts as a physical shield against those hydroxyl radicals. And when researchers at MIT and elsewhere engineered human cells in a lab to express this tardigrade D-soup protein, those human cells suddenly survived 40% more X-ray radiation.
40%. That is a massive biological upgrade, and the delivery method here is key. The text points out that researchers figured out how to deliver the instructions for this D-soup protein using synthetic mRNA packed into lipid nanoparticles.
Right, the same basic tech used in the COVID vaccines.
Yeah. But why mRNA instead of CRISPR for this?
The use of mRNA is a critical distinction here. mRNA is essentially a temporary photocopy of a DNA blueprint. It enters the cell's machinery reads it and manufactures the D-soup protein, and then the mRNA naturally degrades and disappears within a few hours or days.
Ah, so you aren't permanently editing a patient's genome to make them a human tardigrade hybrid.
No, you're not.
You could theoretically give a cancer patient a shot of this mRNA right before radiation therapy. Their healthy cells temporarily manufacture the Kevlar blanket, they survive the massive dose of radiation meant to kill the tumor, and then the shield dissolves a few days later.
Exactly. You get biological enhancement exactly when you need it, and then it's gone.
The clinical applications are obvious, but Enfield immediately extrapolates this to civilizational infrastructure. We're talking about temporary on-demand radiation protection for astronauts on deep space missions.
Or, for emergency workers responding to a nuclear meltdown, we can temporarily borrow the resilience of other species.
But then he goes from temporary shields to, like, permanent regeneration. And he brings up the axolotl, the Mexican walking fish.
Yes, the axolotl.
This is a type of salamander that can have its leg completely bitten off by a predator, and it will just regrow a perfect brand new leg. Bone, muscle, nerves, blood vessels, all seamlessly integrated.
No scarring, just a brand new limb.
How is that even possible?
The mechanism the axolotl uses is called a blastema. When the limb is amputated, the cells at the stump don't form scar tissue like human cells do. Instead, they de-differentiate.
Dedifferentiate.
They revert back to a stem cell-like state, form this mass called a blastema, and then receive organized chemical signals from the surrounding nerves and connective tissue to regrow the exact missing structures.
And here's where Enfield's research absolutely blew my mind. He cites work from Harvard and LSU suggesting that human beings might actually possess the dormant genetic code to do this, like we have the salamander code inside us.
We do. He's referring to the highly conserved genetic toolkits that govern embryonic development. The specific chemical signaling pathways, like the FGF or fibroblast growth factor cascades, which direct tissue formation,
Okay, FGF.
and the or sonic hedgehog pathway, which literally patterns the number and shape of your fingers and toes.
Wait, they named a genetic pathway sonic hedgehog.
They really did. Scientists have a sense of humor.
Yeah.
But these pathways are ancient. They're conserved across vertebrate genomes. You use them when you were a developing embryo to grow your limbs in the first place.
But then, once we are born, evolution just throws a padlock on those folders and suppresses them.
Right. So Enfield's proposition is that we don't need to invent a magic healing potion from scratch. We just need to use a viral nanite to go in, pick the padlock, and flip the transcription factors back on.
We deliver the specific chemical commands that wake up the dormant blastema code.
Exactly. Now, it demands an incredibly complex orchestration of signals in a very specific sequence. You can't just yell, grow at a stump.
Huh.
But theoretically, the raw capacity for complex tissue regeneration hasn't been deleted from the human hard drive. It has merely been relegated to an inactive state to prioritize rapid, crude healing, which is scarring, to prevent fatal infections.
Okay, I have to step in and push back here because this sounds incredibly dangerous. Let's think about the mechanics of this. We send viral nanites into an amputated arm. They start flipping genetic switches, overriding the body's natural suppressors, and commanding human cells to rapidly de-differentiate and explosively multiply to build a new arm.
I see where you're going with this.
I mean, rapid, uncontrolled cellular growth and a refusal to stop divide, that is literally the exact clinical definition of a malignant tumor.
Yeah.
If we tell the cells to act like a salamander, aren't we just guaranteeing a massive aggressive bone cancer at the stump?
You've identified the single greatest hurdle in regenerative medicine, and Enfield actually addresses it head-on. Uncontrolled expression of embryonic growth pathways is a guaranteed recipe for oncology.
Yeah, it has to be.
An embryo and a tumor share a terrifying amount of similar cellular logic. This is why Enfield insists that deploying these biological tools requires an absolute unyielding adherence to a strict three-mode architecture for the nanites.
Okay, break that architecture down for me.
The first mode is seek and destroy. The viral nanite must be programmed to recognize the specific genetic and chemical signature of the damaged, necrotic, or diseased tissue and eliminate it, basically clearing the site.
Makes sense, like clearing the construction site.
Right. The second mode is restore. This is the delicate part, activating the FGF and pathways in the healthy boundary tissue to initiate the organized blastema growth.
But there's a third mode.
Yes. The absolute non-negotiable requirement for this technology to exist safely is the third mode, the built-in genetic kill switch.
So the nanite has an auto-destruct sequence to make sure it doesn't keep running the grow command forever. How do you build a kill switch into a microscopic biological machine?
You engineer synthetic promoter sequences. A promoter is a region of DNA that initiates the transcription of a particular gene. Synthetic biologists can design these promoters to be conditional.
Conditional how?
For example, the nanite might be programmed with a time-limited replication window. It can only execute its instructions for a maximum of 20 cell division cycles, after which the promoter simply degrades.
Wow, okay.
Alternatively, it can be tied to a chemical dependency. The nanite only functions as long as the patient is taking a very specific, rare synthetic molecule as a pill.
Oh, that's clever.
Right. The moment you stop taking the pill, The necessary chemical trigger vanishes, the transcription halts, and the synthetic machinery permanently shuts down.
It's a biological dead man switch. Without the constant signal, the code stops running.
Exactly.
So, to summarize this first massive shift, we are learning to write new code for the software of the human body. We're using stripped-down viruses as highly advanced delivery trucks, and we're architecting built-in kill switches to keep the code from turning into cancer.
That's the foundation, yes.
That alone is a civilizational leap. But what happens if we take that exact same biological logic, the precision, the self-assembly, the programmability, and we stop looking inward at the body and start looking outward at the world?
Right, taking biology to the built environment.
What if we use biology to build physical hardware?
The transition from biological software to biological hardware brings us to the second major pillar of Enfield's framework, Prion and amyloid architectures as the universal material.
Okay, when most people hear the word prion, there is an immediate, visceral reaction of dread. We associate it almost exclusively with mad cow disease or Creutzfeldt-Jakob disease in humans.
Oh, sure.
Like terrifying, untreatable, invariably fatal conditions where your brain essentially turns into a sponge.
The pathology of prion diseases is absolute nightmare fuel. But Enfield's framework requires us to perform a radical conceptual separation.
A separation between the disease and what it's made of.
Exactly. He asks us to strip away the disease context entirely and focus solely on the physical mechanism of what a prion actually is and what it is doing at the molecular level.
Right, because a prion isn't a virus or a bacteria. It doesn't have DNA. It's literally just a protein that has folded into a very specific, stubborn shape.
That's all it is.
And when it bumps into a normal protein, it somehow forces that normal protein to unfold and refold into the exact same stubborn shape. Enfield calls this resulting structure the cross-beta amyloid scaffold.
Right. To visualize this, think of a normal protein as a highly complex piece of origami, folded perfectly to perform a specific job in the cell. A prion is a piece of origami that has been flattened and bent into a sort of Lego brick.
Okay, a Lego brick.
And when this Lego brick touches a normal piece of origami, it forces the origami to flatten out and snap onto the brick. This happens over and over, creating long, incredibly dense fibers.
Ah, I see.
This structure is the cross-beta architecture. It consists of beta strands oriented perpendicular to the long axis of the fiber, locked together by an immense network of hydrogen bonds.
And what materials scientists have realized recently is that this misfolded biological structure, this thing that causes disease, is actually one of the most mechanically robust, information-dense, and tunable nanostructures known to human science.
It's like nature accidentally invented a supermaterial.
The engineering potential is staggering because the material is fundamentally tunable. Enfield highlights a concept called hydrogen bond density, or HBD.
Yes, HBD. This is the engineer's control knob. By altering the specific amino acid sequence of the protein we start with, we can control how tightly those beta strands pack together.
So more bonds means it's stronger.
Exactly. If you increase the hydrogen bond density through tight molecular packing, you yield an extremely stiff, unyielding structural fiber. If you decrease the HBD, introducing more bulky amino acids that disrupt the perfect packing, you get a fragile, highly dynamic structure.
So you are using the exact same underlying protein logic, but you can dial it up to act like structural steel or dial it down to act like a flexible, sensitive biological sensor.
It's completely customizable.
Let's talk about the structural steel end of that spectrum, because the mechanical strength of this stuff is absurd. Enfield cites studies showing that engineered polymeric amyloids boast a toughness of 160 megajoules per cubic meter.
That is a massive number.
For context, that officially beats the toughness of natural spider silk, which has been the holy grail of biomaterials for decades.
And it achieves that extraordinary toughness at roughly 1/8 the density of steel.
How does it do that?
The reason it's so tough lies in those hydrogen bonds. When you apply mechanical stress to an amyloid fiber, say, you try to stretch it or bend it, the massive network of hydrogen bonds begins to break and reform.
Like continuously.
Continuously. Breaking and reforming, continuously dissipating the kinetic energy of the stress before the actual covalent backbone of the protein snaps. It's a self-healing shock absorber at the nanoscale.
A biocompatible, ultra-tough nanomaterial made out of protein. But its physical strength is just the baseline. This is where the physics gets incredibly weird. Ships and colleagues demonstrated in 2021 that these amyloid crystals aren't just strong.
No, they can actually conduct electrons.
Right. They exhibit conductivity through a phenomenon called tyrosine hopping.
Yeah, tyrosine hopping. Proteins are made of amino acids, right? Tyrosine is an amino acid that contains an aromatic ring, a molecular structure shaped like a hexagon with delocalized electrons buzzing around it.
Buzzing around it, okay.
When the cross-beta scaffold forces these tyrosine rings into a perfectly stacked alignment, their electron clouds overlap. This creates an orbital pathway, an invisible highway, that allows an electron to physically hop from one tyrosine ring to the next over micrometer distances.
It's an intrinsic biological nanowire. You don't need copper, you don't need heavy metals, you don't need toxic silicon foundries. The protein just lines up and conducts electricity.
That's wild.
But wait, it gets even more unapologetically sci-fi. Enfield notes that these materials are also highly piezoelectric.
Yes, piezoelectricity is a phenomenon where a material generates an electrical charge in response to applied mechanical stress.
So like the spark clicker on a gas grill. You apply physical pressure to a crystal, and it snaps an electric spark.
That's the exact mechanism.
Yeah.
Conversely, if you apply an electrical field to the material, it physically deforms or vibrates.
And these engineered amyloids have a piezoelectric constant of 24 to 62 picometers per volt. Enfield points out that this rivals synthetic petroleum-based polymers like PVDF, but the amyloids can be manufactured from cheap food-grade proteins like lysozyme or insulin.
The applications for a biological piezoelectric material are revolutionary, particularly in medicine. Enfield highlights a 2024 study by Dolai and colleagues that demonstrated a fully self-contained therapeutic transducer.
Okay, break that down.
Imagine injecting a solution of these amyloid fibrils directly into a solid tumor. Because they are piezoelectric, they act as acoustic to electrical converters.
So you inject the tumor, and then you hit the patient with external ultrasound waves, sound waves.
Precisely. The acoustic pressure waves from the ultrasound physically squeeze the amyloid fibrils. Because they are piezoelectric, that physical squeezing generates localized electrical charges right there inside the tumor.
Wow.
Those electrical charges interact with the water and oxygen in the tissue to create reactive oxygen species.
Which are highly toxic molecules that shred the cell walls and DNA of the surrounding cancer cells.
Exactly.
You are literally zapping the tumor from the inside out, using harmless sound waves from the outside to power a biological battery on the inside.
It bypasses the need for systemic chemotherapy entirely.
That is just wow. But even killing tumors pales in comparison to the most radical application Enfield proposes for these conductive piezoelectric fibers, DNA prion computing.
This is where we start talking about the next era of information technology.
Okay, this blew my mind. Standard digital computing, the device you are listening to this on right now, is binary. It uses silicon transistors to represent ones and zeros. But Enfield is proposing we build processors using the quaternary code of DNA.
Yes, DNA fundamentally processes information using 4 distinct chemical bases: adenine, cytosine, thymine, and guanine, ACTG.
Right.
That means every single molecular bit of DNA has 4 possible states, not 2. The information density is almost incomprehensible. Theoretically, a single gram of DNA can store over 200 petabytes of data.
All the data on the internet could fit in a shoebox.
Easily, but the bottleneck for DNA computing has always been speed and wiring. How do you read and write data to a DNA molecule quickly? And how do you wire billions of DNA molecules together to form a processor without using clunky silicon interfaces that totally defeat the purpose of going microscopic?
Exactly. How do you connect it all?
Enfield synthesis solves both bottlenecks. To wire the DNA memory sites together, you use the conductive prion fibers we just discussed. They naturally self-assemble into intricate branching nanowire networks.
So they build their own wires.
Yes. Furthermore, because their physical shape depends on environmental factors, they can be engineered to act as physical logic gates, routing electrical signals based on their conformation.
And for the speed, how do you clock a biological processor?
Enfield proposes using terahertz radiation. The hydrogen bonds that hold the 2 strands of the DNA double helix together naturally vibrate at terahertz frequencies.
Okay.
By hitting the DNA with highly focused, intense terahertz pulses, you can resonantly excite those bonds. This causes the DNA strands to rapidly unzip, exposing the code, and rezip in picoseconds.
So the terahertz pulses act as the hyperfast clock speed for the DNA memory, while the prion fibers act as the conductive interconnects.
Exactly. You are describing a processor unimaginably denser and faster than silicon, and it is completely grown from a biological soup.
Grown from a biological soup. Let's hover on the manufacturing side of this for a minute, because this sounds both amazing and terrifying. The analogy I kept coming back to for prion self-assembly is a zombie outbreak, but for industrial manufacturing.
I like that analogy.
If you want to make a batch of these biological nanowires, you don't need a multi-billion-dollar fabrication plant with clean rooms and vacuum chambers. You just take a vat of normal liquid protein soup and you drop in one single seed fiber, the zombie.
The templating cascade. The moment that single structural seed enters the solution, it begins recruiting the normal proteins, forcing them to misfold into identical copies of itself.
And then it just spreads.
Yep. Those copies then recruit more proteins in an exponentially expanding chain reaction until the entire vat of liquid protein has been converted into perfect, solid structural fibers.
The energy cost of manufacturing drops essentially to zero. It happens at room temperature. But I have a massive pushback here.
Okay, let's hear it.
We keep talking about prions. Using the literal physical mechanism behind mad cow disease to build spaceship hulls or medical sensors or the computer chips in our phones sounds like an extinction-level horror movie waiting to happen.
It does sound sketchy on the surface.
Right. If a microscopic flake of my biological computer chip breaks off and I inhale it, or if a piece of our space station hull scratches a crew member, how do we ensure that templating cascade doesn't accidentally trigger inside a human brain and start turning our own proteins into zombie fibers?
The containment and biosafety issue is paramount. It is the first question any rational observer should ask. However, Enfield makes a crucial foundational distinction in the text that separates the engineering material from the pathogen.
Okay, what is it?
The proteins utilized for these industrial and computing applications are absolutely not the mammalian PRP prion that causes Creutzfeldt-Jakob or mad cow disease.
Okay, if they aren't human prions, what are they?
They are based on yeast prion determinants, specifically proteins like sub-35, or they are entirely synthetic peptides engineered from scratch in a laboratory.
But they still misfold, right? They still do the zombie cascade.
They possess the structural logic of prion self-assembly. They have the ability to form the rigid cross-beta scaffold. But, and this is the critical safeguard, they possess zero infectivity to humans.
Why?
For a prion cascade to jump from an engineered material into a human body, the engineered prion must share a high degree of sequence homology with a human protein. It needs a target that is chemically similar enough to latch onto and force to misfold.
I see. So it needs the right kind of raw material to continue the cascade.
Exactly. And the amino acid sequences of these synthetic and yeast-derived prions simply do not exist in the human proteome.
So what happens if you get exposed to it?
If you inhaled a flake of an engineered yeast prion, your body's proteases would simply break it down like any other foreign protein you might eat in a piece of bread. It is chemically blind to human biology. It borrows the geometric mechanism of the cascade, but it completely isolates the pathology.
Okay, that significantly lowers my blood pressure. So to recap where we are, we have this miraculous, tunable, super strong, conductive, self-assembling material, and it's relatively safe because it ignores human proteins. But here's the next huge problem. A zombie cascade in a vat of liquid just makes a chaotic, tangled mess of microscopic fibers. It grows in every direction at once. If it's just a tangled ball of biological wire, how do we actually shape it? How do we take a vat of fluid and force it to grow into the exact shape of a computer chip or an artificial organ or a structural panel?
You've highlighted the inherent limitation of simple amyloid engineering. It lacks spatial programmability. You can start the reaction, but you can't tell it where to go.
Right.
Overcoming this limitation is the focus of the third section of Enfield's architecture, which he calls Prion optogenetic lithography.
Writing with light. This is where Enfield proposes taking three existing radically different technologies and smashing them together into one unified system.
Yes.
We have the synthetic prion self-assembly we just talked about. We combine that with optogenetics, and then we combine both of those with photolithography.
Let's define the components. Optogenetics is a field well established in neuroscience. Researchers identified specific proteins in plants and algae, like the LOV2 domain or the Cry2 protein, that respond dynamically to light.
Because plants need to know where the sun is.
Exactly. So they have proteins that literally change their physical shape when struck by very specific wavelengths of photons.
When the light hits them, the energy from the photons causes the chemical bonds in the protein to flex and twist. It's like a molecular lock and key, but the key is a beam of light.
Precisely. And photolithography is the technique the semiconductor industry uses today. They use highly focused lasers to draw microscopic circuit patterns onto silicon wafers.
Okay, so how do they combine?
Enfield's brilliant synthesis is to fuse these concepts at the molecular level. Imagine a synthetic prion protein. On its own, it wants to cascade and form fibers. But scientists genetically fuse an optogenetic lock protein like that LOV2 domain directly onto the prion.
And this is where the system becomes programmable. As long as the vat of protein soup is kept in the dark, the optogenetic protein acts like a cage. Yes.
It wraps around the prion sequence, physically hiding it. The protein can't cascade, the liquid just sits there completely inert.
But the exact microsecond, a highly focused, specific wavelength laser beam penetrates the liquid and hits that molecule.
The photon energy twists the lock. The cage opens. The prion sequence is suddenly exposed, and the solid fiber instantly begins to grow. But it only grows precisely in the microscopic physical space where the laser beam is touching the liquid.
It completely solves the spatial control problem. You aren't just dropping a seed into a vat and hoping for the best. You are using overlapping holographic beams of light to create localized points of intensity within a three-dimensional vat of liquid.
So wherever the beams cross,
The light intensity crosses the threshold, the locks open, the cascade triggers, and the liquid freezes into a solid cross-beta structure midair.
here's where it gets really interesting. It is essentially a 3D printer, but it has no moving parts. There is no mechanical nozzle extruding a paste.
None at all. The printer is just a clear tank of biological fluid, and lasers are projecting a three-dimensional hologram into the tank, and the object just materializes in the fluid drawn by light.
You are writing three-dimensional protein architecture with molecular precision. And the applications and field outlines for this optogenetic lithography are paradigm shifting. Let's look at a seemingly mundane but globally critical application.
Food printing.
Yes, the text dives deep into printing meat, because right now, the plant-based meat industry relies on extruding mushy soy or pea protein into patties. They can't replicate a steak because a steak has complex microscopic structure.
Exactly. A steak is a highly organized architecture of aligned muscle fiber bundles interlaced with a precise matrix of connective tissue and fat cells. Extrusion cannot recreate that microarchitecture.
But optogenetic lithography can. The text details how you could print a meat analog, voxel by voxel.
A voxel being a three-dimensional pixel.
Right. Imagine a massive, clear bioreactor vat filled with photosynthetically grown liquid protein feedstock, perhaps sourced from rapidly multiplying algae.
You project patterned light into this vat. The light directs the specific folding and assembly of that protein, writing the dense, aligned fiber bundles of the muscle tissue, and then shifting the light pattern slightly to write the softer, cross-linked matrix of the connective tissue around it.
You are literally growing the structural snap of a shrimp shell or the tough chew of a beefsteak at the micrometer scale. You create the exact physical experience of meat, but with zero animals slaughtered, zero antibiotics used, and a fraction of the land and water required.
It's just patterned light organizing algae protein.
And if you can organize protein to simulate muscle for food, you can organize it to simulate tissue for medicine. Enfield transitions the logic to neural scaffolding.
Oh, this was amazing.
Consider a patient suffering from a severed spinal cord or severe traumatic brain injury. The central nervous system is notoriously bad at healing. If you just inject a healing hydrogel into a brain lesion, the regenerating neurons just wander around aimlessly. They cannot bridge a large gap.
They need a physical path to follow, like a trellis.
Exactly. So Enfield suggests using implanted microscopic fiber optics to project a three-dimensional hologram of light directly into the damaged area of the brain or spine.
You inject the inert, optogenetically locked prion solution into the spinal gap. Then you activate the fiber optics. The light projects the exact pre-calculated geometry of the lost white matter tract.
And instantly, a physical, solid protein scaffold materializes in the gap, perfectly matching the original neural architecture. The regenerating axons now have a physical trellis to grow along, allowing them to cross the lesion and reconnect the circuit.
It's breathtaking. You are projecting a ghost of the missing nervous system, and biology materializes to fill it. And obviously this applies to technology as well. Enfield calls it in situ circuitry.
Yes. Instead of building a microchip in a multi-billion-dollar clean room using highly toxic etching chemicals and extreme heat, you can literally write conductive amyloid nanocircuits directly into a liquid substrate.
You can print complex sensors or biological processors on the fly at room temperature using light. It shifts global manufacturing from a subtractive, energy-intensive process into an additive, biologically compatible one.
It's a total revolution in manufacturing.
But I'm going to play devil's advocate again here. Let's think about the real-world safety of this. Let's say I buy an optogenetically printed steak or a biological computer chip and I take it out of the lab. What happens if I expose it to ambient sunlight?
I see we go.
The sun emits a massive spectrum of UV and visible light. Doesn't the sun just act like a giant laser, unlocking all the remaining caged proteins in the structure, causing the thing to relentlessly cascade and grow endlessly until it consumes my entire kitchen?
It's a vivid and horrifying image, but Enfield details the safety protocols that make that scenario impossible. He mandates a strict two-level kill switch system for optogenetic materials.
What's the first level?
The first level is simply the physical limitation of monomer supply.
Monomers being the individual, unattached proteins floating in the liquid.
Correct. The structure can only grow as long as there is free liquid protein available to feed the templating cascade. The cascade consumes its environment.
What?
Once the printing process is finished, you remove the solid object from the vat and wash away any residual liquid. Even if you blast the solid object with perfect UV light, it cannot grow because it has no fuel. There's nothing left to convert.
Okay, that makes sense. A fire can't burn without wood, no matter how much you spark it. But what about inside the vat? What if the laser misfires and starts a runaway reaction in the tank?
That is where the second level of control comes in. Enfield references advanced optogenetic tools developed at Texas A&M, specifically the PHOBE-1 systems. These systems provide light-off reversibility.
Light-off reversibility.
You engineer the molecular lock so that one specific wavelength of light, say blue light, causes the protein to unfold and the cascade to begin. But an entirely different wavelength of light, say far red light, causes the protein to forcibly snap back into its locked, inactive state.
So if a reaction starts growing out of control, you just flood the entire vat with red light, and every single lock instantly slams shut, halting the cascade immediately.
Exactly. It is a fully reversible, tightly controlled thermodynamic switch.
Okay, so we've zoomed incredibly far in. We've talked about writing software in DNA, using viral delivery trucks, and shooting lasers into vats of liquid to print structural hardware out of protein.
Right.
We are talking about a civilization that relies on vast oceans of biological fluid and constant cellular manufacturing. But to make this entire vision work, Enfield demands we zoom all the way out. Which brings us to the fourth major pillar of his architecture. From the ground up, planetary citizenship and infrastructure.
This is a huge pivot.
Because if we are going to print cities, food, and computers out of biological matter, how do we produce the massive, staggering amounts of protein and energy needed to sustain this civilization without completely destroying the planet in the process?
This is the crucial pivot in the text. Enfield shifts from the extreme micro scale of molecular engineering to the macro scale of civilizational ethics and ecological management. He argues that our current industrial civilization suffers from a fatal flaw that he calls the linear fallacy.
The linear fallacy, meaning a straight line from extraction to destruction.
Exactly. We extract a resource from the earth, we process it using intense heat and pressure, we utilize it briefly, and then we discard the waste into the environment. We pull fossil water from an ancient aquifer, we synthesize nitrogen fertilizer using natural gas, we dump it on a field to grow corn, and we allow the massive excess of those nutrients to wash into our river systems.
And that runoff flows down the Mississippi River, dumps into the Gulf of Mexico, triggers massive algae blooms that suck all the oxygen out of the water, and creates oceanic dead zones the size of New Jersey where nothing can live.
It is a one-way trip to maximum entropy. The linear system assumes the Earth is an infinite piggy bank on one end and an infinite trash can on the other.
Which obviously isn't true.
No. But biological systems have been perfecting resource cycling for 3.5 billion years. In a mature ecosystem, there is no such thing as waste. The byproduct of one process is the essential fuel for another.
Right.
Enfield argues that if we want to survive our climate and ecological crises, we must adopt biological circularity at the landscape level. We have to stop looking at plants and microbes as fragile nature that needs to be cordoned off in national parks and start engineering them as hard civilizational infrastructure.
He says that instead of seeing waste as a disposal problem, we have to recognize it as misplaced fertility. I was trying to come up with an analogy for this.
Let's hear.
Our current industrial system is basically like trying to run a household by just continuously throwing your garbage and your raw sewage out the living room window into your neighbor's yard.
Gross, but accurate.
Right. Eventually, you run out of things inside your house, and the outside of your house becomes an unlivable, toxic wasteland. But Enfield's biological loop is like turning the entire planet into the ultimate self-powering, perfectly balanced compost bin.
That is precisely the paradigm shift he is advocating. And he doesn't just philosophize. He provides concrete, mathematically modeled blueprints for this infrastructure. The first major framework he explores is bio-CCS, or biological carbon capture and storage. He focuses heavily on industrial-scale algae scrubbers.
Right. Because if you want to feed all those 3D light printers we just talked about, you need an unfathomable amount of liquid protein feedstock. You need a way to grow biomass incredibly fast.
And when it comes to rapid biomass generation, microalgae are unmatched. Enfield cites figures showing that certain strains of microalgae can produce 10,000 gallons of lipid oil per acre per year.
That sounds like a lot.
Compare that to our traditional agricultural champion, soybeans, which produce roughly 48 gallons of oil per acre per year.
Oh, wow. 10,000 compared to 48.
It is a transformative difference in efficiency. But the genius of bio-CCS is how you feed the algae. You don't use clean water and pristine fertilizer. You pump the raw, toxic exhaust flue gas directly from coal-fired power plants or ethanol refineries into the algae bioreactors.
You literally feed the atmospheric pollution to the algae.
Yes. The intense concentration of CO2 in the industrial exhaust supercharges the photosynthetic growth of the algae.
So you are taking a toxic carbon waste stream that was going to choke the atmosphere, and you're using it to rapidly synthesize harvestable biodiesel and high-quality protein feedstock.
The pollution ceases to be a liability and becomes an essential, monetized nutrient stream. And Enfield applies this exact same logic of circularity to large-scale agriculture with a framework he calls the duckweed loop.
This section was absolutely fascinating. Duckweed. That tiny, bright green floating plant you see covering the surface of stagnant ponds and slow-moving creeks.
Memnoidea, or duckweed, is the fastest-growing vascular plant on Earth. Under optimal conditions, duckweed can double its entire biomass every 16 to 48 hours.
It is an explosive grower.
And crucially, it thrives on the exact chemical compounds that cause our agricultural pollution crises: nitrogen and phosphorus.
So Enfield says, instead of letting all that excess synthetic fertilizer runoff from Midwestern corn and soy fields wash into the Mississippi River and cause the dead zones, you intercept it.
You channel that agricultural runoff into massively scaled, shallow, managed duckweed ponds built along the edges of the fields.
The duckweed acts as an aggressive biological filter. As the runoff flows through the pond system, the rapidly multiplying duckweed strips up to 96% of the phosphorus and 67% of the nitrogen right out of the water.
It biologically remediates the water, largely for free, before it ever reaches the river. But that is only the first half of the loop.
The second half is the harvest. Because duckweed isn't just a filter, it's a nutritional powerhouse. The harvested dried weed packs roughly 33 to 44% raw protein,
Which is nutritionally competitive with highly intensive crops like alfalfa and soy. So the farmer harvests the duckweed from the filtration ponds. They can dry it and apply it back onto the fields as a slow-release, free, organic fertilizer, replacing the need to buy synthetic chemicals next season.
or they can process it into high-protein feed for their livestock.
Exactly. The farmer stops purchasing expensive synthetic inputs, stops paying fines for polluting the watershed, cleans the local ecosystem, and generates a free, continuous supply of livestock feed.
It's the ultimate closed loop. But Enfield notes that we are currently doing the exact opposite. He points to a very specific, slow-moving disaster in Nebraska to illustrate why we desperately need this shift in thinking. The Ogallala Aquifer crisis.
The Ogallala Aquifer is one of the largest underground freshwater reserves in the world. It spans beneath the Great Plains and is the lifeblood of American agriculture. It is a geologic fossil resource. It took millions of years of glacial meltwater to fill, and through relentless mechanized pumping, we are draining it dry in a matter of decades.
That's terrifying.
Portions of the aquifer in Kansas and Texas are already effectively exhausted.
But the truly fascinating and tragic part of Enfield's analysis is what he calls the efficiency paradox of modern agriculture. He looks at the history of how farmers in Nebraska used to irrigate. Decades ago, they relied on gravity-fed dirt ditches. They would pump water from the aquifer and just let it run down unlined trenches between the crops.
By any modern engineering metric, it was terribly inefficient. The dirt ditches leaked water profusely into the ground. They created muddy, unusable wetlands at the edges of fields. And being completely exposed to the sun and wind, they lost massive volumes of water to evaporation.
So, wanting to conserve water and maximize yield, the agricultural industry heavily subsidized and adopted center pivot sprinklers. Those massive, alien-looking metal arms on wheels that you see flying over the Midwest when you look out an airplane window.
They are highly pressurized, incredibly precise, and mathematically efficient. They drop the exact required amount of water directly onto the root zone of the plant.
But the paradox, which is a classic manifestation of Jevons paradox in economics, is this. When the irrigation technology became highly efficient, farmers did not pump less water from the aquifer.
No, they didn't.
The water savings simply made irrigation cheaper. So they used the saved water to expand their operations and irrigate significantly more acres of marginal land. The total draw on the aquifer actually accelerated. And meanwhile, because they had the sprinklers, they plowed over, paved, or abandoned all of those old, leaky, inefficient dirt ditches.
And this is where the unseen tragedy occurs. Enfield points out that those old, inefficient dirt ditches were secretly performing two massive, unrecognized ecological jobs. First, all that leaky water seeping into the mud was actually slowly percolating back down through the soil and naturally recharging the aquifer.
Yes, the ditches acted as inadvertent, massive infiltration galleries. They were ecological infrastructure hiding in plain sight.
And second, the massive evaporation that the engineers hated. All that standing water evaporating into the hot, dry Nebraska summer air was significantly increasing the local humidity. It was putting so much moisture back into the boundary layer of the atmosphere that it actually provided the necessary latent heat and moisture to trigger localized summer thunderstorms.
By upgrading to efficient closed pipes and sprinklers, we accidentally stopped the natural aquifer recharge, and we literally stopped the rain.
This forces a profound reevaluation of how industrial civilization defines efficiency. An efficiency metric that isolates a single variable like crop yield per gallon of pumped water, while entirely ignoring and ultimately destroying the underlying hydrologic cycle that sustains the entire region, is a catastrophic failure of systems thinking.
That is why Enfield proposes what he calls Ditch 2.0.
Right.
He argues we need to purposefully bring the ditches back, not the old sloppy ones, but managed, highly engineered biological corridors.
Precisely. Ditch 2.0 combines the duckweed loop with hydrologic engineering. You build vast networks of shallow canals, they intercept the fertilizer runoff, they grow the duckweed to scrub the nitrogen and harvest the protein.
And the massive surface area deliberately evaporates moisture back into the atmosphere to restore the localized precipitation cycles.
And the subsurface of the canals is engineered with specific gravel and sand gradients to deliberately, slowly filter and recharge the clean water back into the Ogalla aquifer.
You rebuild the kidneys of the landscape. It sounds beautiful. It paints this incredibly idyllic picture of a perfectly balanced, self-sustaining Midwestern farm. But I have to stop you and push back hard on the reality of this.
Let's do it.
Let's look at the actual scale of the American Midwest. We are talking about millions upon millions of tons of concentrated, highly toxic synthetic fertilizer runoff. If you concentrate all of that into stagnant, shallow, warm water trenches around every farm, duckweed isn't the only thing that loves nitrogen.
Cyanobacteria loves it too.
Right, the blue-green algae, the stuff that causes highly toxic microcystin blooms that kill dogs who swim in it and shut down city water supplies. Are we really just cleaning the water, or are we accidentally engineering thousands of miles of toxic, microcystin-filled hazard zones in the backyards of rural America?
That's a very fair concern.
And furthermore, what happens during a massive spring flood? A torrential downpour hits Nebraska. The treatment ponds overflow their banks, the toxic water washes into the clean livestock ponds, and suddenly you've poisoned the entire regional food supply.
Those are highly critical, pragmatic engineering concerns. And to be clear, Enfield's text does not gloss over the severe biological risks of scaling open-air phytoremediation. He addresses the cyanobacteria threat by breaking down the necessary operational requirements and the underlying stoichiometry.
Okay, unpack stoichiometry for me in this context.
In chemistry, stoichiometry is the calculation of reactants and products in a chemical reaction. You have to ensure the math balances. In this context, it means you cannot just dig a hole, fill it with runoff, throw some duckweed in, and hope for the best. You must precisely calculate the massive tonnage of incoming nitrogen and ensure the physical volume of the duckweed pond and the harvesting rate mathematically exceed the nutrient load. If the duckweed dominates the nutrient uptake, it outcompetes the cyanobacteria. starving the toxic blooms before they form.
It has to be an actively managed biological machine, not just a passive pond.
Exactly. But to address your point about flooding and contamination, Enfield insists on strict physical, civil engineering separation. You establish two entirely disconnected systems: treatment biomass and feed biomass.
Walk me through the separation.
Treatment biomass refers to the duckweed grown in the primary interceptor canals, the trenches receiving the raw, heavy agricultural runoff, or even municipal sewage. Its sole job is rapid extraction of heavy pollutants and heavy metals. You absolutely do not feed this biomass to animals.
So what do you do with it?
You harvest it, and you run it through massive, sealed anaerobic digesters. The digesters use specialized bacteria to break down the toxic plant matter, generating methane biogas to power the farm's electrical grid, while the intense heat and bacterial action destroy the pathogens and microsystems.
And the remaining sterile sludge is then safely composted into inert soil amendment.
Exactly.
Okay, so the dirty stuff is processed for energy and sterilized. Where does the animal feed come from?
From the completely isolated feed biomass systems. These are separate closed-loop ponds grown on clean, controlled nutrient streams, perhaps utilizing the sterile, composted output from the digesters. This duckweed is what is harvested and processed for livestock feed.
And what about the flood risk?
Massive berms, overflow spillways, and redundant geographic elevation drops are engineered into the landscape to ensure that even a 100-year flood event physically prevents the treatment canals from backflowing into the feed ponds. It requires treating the biological landscape with the exact same rigorous safety tolerances and massive civil engineering investment that we currently apply to chemical treatment plants or oil refineries.
It's a massive infrastructure project. And crucially, looking impartially at the text, Enfield points out that the reason this biological circularity isn't already standard practice everywhere isn't because the science is faulty or the engineering is impossible. It is because our current economic and subsidy structures actively punish it.
Yes. The text emphasizes, and to be clear, the author is conducting an economic analysis here, not taking a political or partisan stance, that the current agricultural tax incentives and federal subsidy frameworks systematically reward farmers based almost entirely on maximum volume production.
So just grow as much as possible, regardless of the cost to the environment.
Exactly. Maximizing raw volume requires maximizing chemical inputs and aggressive water extraction. The negative externalities of that process, the depleted aquifer, the poisoned river, the oceanic dead zones, are not financially priced into the agricultural market.
So the linear destructive system appears mathematically more profitable on a quarterly balance sheet, even though it is fundamentally liquidating its own foundational natural capital.
It's a failure of systems accounting, not a failure of biology.
Okay, so this brings us to the final and arguably most intense part of our deep dive. The framework Enfield lays out is undeniably beautiful, a world where viruses cure disease permanently, where lasers print structural material from misfolded proteins, where our farms actively repair the atmosphere and the aquifers.
It's a very compelling vision.
But the sheer power to rewrite human DNA, to invent new biological machinery, and to fundamentally alter planetary ecosystems introduces terrifying existential risks. Which brings us to Enfield's final essay, The Ethics of Programmable Evolution. What are the rules of this new world?
Enfield centers the ethics of this entire transition on a single, uncompromising concept: planetary citizenship. It is the philosophical realization that living on a biologically active, living planet requires reciprocal maintenance, not just blind extraction.
If you consume the ecological services of the Earth clean water, a stable climate, the genetic repository of biological diversity, you inherit an absolute obligation to maintain and replenish them.
Right.
So what does this all mean for us as a species? Because listening to this, it sounds like we are essentially handing humanity the administrative passwords to the operating system of life on Earth.
We are crossing a Rubicon. We are moving from millions of years of blind, random Darwinian evolution to an era of intentional, directed biological authorship. And Enfield is very clear that the risks of doing this are astronomical.
Yeah, they have to be.
But what is truly fascinating is that he argues the greatest immediate threat we face in this transition isn't what Hollywood expects. It isn't an engineered biological weapon or a synthetic supervirus escaping a lab.
No. If it's not a supervirus, what is the ultimate threat?
It is the threat of a permanent biological class divide.
Oh, right. If we look back at the CRISPR therapies we talked about in the very beginning, Casgevy, the miraculous permanent cure for sickle cell disease, Enfield notes in the text that the initial price tag for that single treatment was $2.2 million per patient.
Exactly. The foundational therapies of this new era are staggeringly expensive. Now, extrapolate that curve. Imagine the moment when the technology inevitably moves beyond simply curing diseases to actual elective biological enhancement.
Enhanced radiation resistance for space travel, extended visual spectrums.
Increased oxygen efficiency allowing for superhuman physical endurance. If those biological upgrades become luxury commodities governed purely by the free market, available only to the ultra-wealthy, we risk a horrifying scenario of permanent physiological stratification.
We risk a literal biological speciation event, splitting humanity into the enhanced rich and the baseline poor.
The wealthy become a practically superhuman species, immune to cancer and radiation, while the poor are stuck with baseline biology, suffering from the very environmental collapse the linear economy created. It is profoundly dystopian.
Which is exactly why Enfield argues, with extreme urgency, that access to biological enhancement must be treated fundamentally as a public health utility, not a luxury consumer market.
And to prevent this speciation and to guard against biological catastrophe, he lays out strict, non-negotiable ethical architectures. For instance, he draws a massive hard line in the sand between two types of genetic engineering: somatic edits and germline edits.
Break that distinction down for me because it's crucial.
A somatic edit targets the non-reproductive cells in your body. If you receive a viral vector therapy to fix the code in your liver, it only fixes your liver. The edit dies with you.
Okay, that makes sense.
A germline edit, however, targets the reproductive cells, the sperm, the egg, or the early-stage embryo. If you make a germline edit, that genetic change is permanently written into the lineage. It is passed down to every single descendant of that person, forever.
Like the highly controversial 2018 case he highlights in the text, where the biophysicist He-Jiang Cui illegally edited the CCR5 gene in human embryos to try and make the resulting twin girls immune to HIV.
Precisely. That experiment caused universal international outrage across the scientific community because those children could not possibly consent to having their genome permanently altered, and the long-term, multi-generational cascade effects of turning off that receptor are entirely unknown.
Enfield argues that there must be an ironclad permanent global moratorium on all germline edits. We cannot write experimental code into future generations. We must strictly limit ourselves to somatic edits where a living, breathing adult can understand the risks and give informed consent for their own individual body.
And another massive ethical pillar he demands is reversibility. If we are going to enhance human biology, we must always maintain the ability to hit the undo button.
Consider the MIT tardigrade study we discussed. The researchers did not permanently edit the human genome to express the DSEP shield. They utilized synthetic mRNA to deliver the instructions.
mRNA is inherently unstable. It degrades naturally in the cellular environment within hours or days. Enfield argues that a temporary, highly targeted enhancement like mRNA is ethically vastly superior to a permanent genomic edit. It preserves the individual's fundamental biological autonomy and their right to withdraw consent from the enhancement.
But I am going to push back incredibly hard on this concept of biological control. We sit here and talk about kill switches and reversibility and strict separation as if biology is a perfectly obedient, predictable machine.
It's definitely not.
Right, as if we are just pressing buttons on a sterile silicon keyboard. But life's entire defining trait, the entire mechanism of evolution for the last 4 billion years, is mutation, adaptation, and relentless survival. Life finds a way to break the rules. Can we really hubristically guarantee that a synthetic biological kill switch won't just experience a random mutation, adapt, and evolve to completely ignore our off signal?
You have zeroed in on what is undoubtedly the ultimate technical, philosophical, and existential challenge of this entire framework. It is the defining risk of programmable biology. Life is inherently noisy, it is unstable, it is profoundly prone to mutation.
So it can fail.
Can a genetic kill switch fail? Yes. Can a synthetic promoter sequence undergo a point mutation that renders it permanently active? Yes. The illusion of absolute control is the greatest danger we face.
So we could theoretically unleash a self-replicating biological machine, a viral nanite, or a synthetic prion that experiences a random mutation, decides it doesn't want to die when the time limit expires, and just continues to replicate endlessly.
That potential failure mode is exactly why Enfield emphasizes that biological safeguards cannot be bureaucratic regulatory afterthoughts. They have to be the very first architectural principles of any design. It underscores exactly why the FDA currently mandates a mandatory 15-year long-term safety and surveillance tracking period for all patients receiving CRISPR gene therapies.
You do not just inject the genetic patch and walk away assuming it worked perfectly. You comprehensively monitor the cellular behavior, looking for off-target edits or runaway mutations for decades.
You have to assume the code will eventually try to break itself.
Exactly. You must build wildly redundant, overlapping systems of failure. You don't just rely on one genetic kill switch. You build a genetic timer that stops replication, and you pair it with a structural dependency, a requirement for a synthetic amino acid that does not exist in nature.
If the genetic timer mutates and fails, the organism still starves to death because it cannot find the synthetic chemical fuel it needs to build its cellular walls. It is an incredibly dangerous, massive risk.
That's it, but Enfield concludes his essays with a sobering counterargument.
He does. He argues the risk of not taking intentional control of our biological destiny, the risk of continuing to let random genetic disease ravage our bodies, and continuing the blind, linear extraction that is actively triggering global ecological collapse is a far worse, guaranteed path to ruin.
It's the ultimate civilizational tightrope walk. We are being forced to step up and become the active authors of our own biology and the stewards of our planetary infrastructure. But we have to do it with a level of extreme humility, foresight, and caution that humanity is not exactly historically famous for.
The underlying code of life is sacred, Enfield suggests, not because it's perfectly immutable and untouchable, but because it is ours. We are made of it. And for the very first time in the history of the Earth, the code is learning how to read and rewrite itself.
That is a staggering amount of information to process. We have covered an immense landscape today, tracing Enfield's architectural vision from the absolute microscopic up to the planetary. We started by fundamentally reframing disease, seeing how viruses, stripped of their deadly payloads, are being repurposed right now as microscopic delivery trucks.
Delivering precise CRISPR patches to debug the corrupted software of our own cells.
We examined how misfolded prions, traditionally the villains of terrifying neurodegenerative diseases, can be conceptually flipped and harnessed as the ultimate, tunable, self-assembling, universal material-yielding, piezoelectric tumor zappers and DNA-powered terahertz supercomputers.
We explored the mind-bending fusion of those prions with optogenetics and photolithography, allowing us to literally write solid physical objects from structured printed meat to complex neural scaffolds out of thin air, using nothing but intersecting beams of laser light in a vat of liquid protein.
And then we zoomed all the way out to the macro scale, confronting the destructive linear fallacy of our modern world. We saw how large-scale biological infrastructure like algae scrubbers feeding on industrial exhaust and massively engineered duckweed loops restoring the ancient Ogalla aquifer can close the loops of our civilization, turning toxic waste back into essential fertility.
But all of this power rests precariously on the ethical foundation of planetary citizenship. We cannot safely wield the power of programmable evolution without fully accepting the absolute responsibility to maintain the ecosystems that sustain it while vigilantly guarding against the nightmare of biological class division.
It is a framework that redefines our relationship with nature, shifting us from conquerors to participants.
It really is.
It's incredible. And to you listening, the most important takeaway I can leave you with is this. This isn't an abstract sci-fi movie set in the year 2300. This is happening right now, today. Casgevy is FDA approved in curing sickle cell. Duckweed ponds are actively filtering municipal water.
You are made of the exact same biological code we've been talking about for the last hour.
The food you eat, the medicine you take, and the very climate you live in are actively transitioning into this programmable paradigm as we speak.
It is a profound, irreversible shift in what it means to be a human being and what it means to physically inhabit this planet.
So as we wrap things up, I want to leave you with one final provocative thought, something we touched on but didn't fully resolve in the text. Enfield talks about planetary citizenship, meaning we have a duty to give back to the ecosystem we consume. Well, imagine a future where you have successfully received one of these biological enhancements.
Okay, picture this.
Let's say you have a synthetic, optogenetically printed amyloid organ implanted in your chest that gives you extended breath-holding capacity or dramatically enhanced stamina. But to power that synthetic biology inside your body, it requires a very specific, complex protein or chemical catalyst that is physically generated by the massive algae scrubbers cleaning the air in your local city's infrastructure.
A literal physical dependency on the health of your local ecological infrastructure.
Exactly. what happens to our concept of self when your very biology is literally functionally plugged into the local ecosystem? You aren't just living in nature anymore, looking at it through a window. Your heartbeat, your physical stamina is inextricably tethered to the chemical health of the duckweed in the ditch down the street.
If the local river gets polluted and the algae dies, you physically, personally feel the biological downgrade in your own body within hours.
Does establishing that immediate physical feedback loop finally force humanity to treat the planet with the respect it deserves, simply because the planet is now literally an extension of our own bodies?
It completely erases the boundary between the individual organism and the global environment.
It sure does. And that is a fascinating, heavy concept for you to ponder. Until next time. Thank you for joining us on this Doc Dive.
Thanks for listening.
See you next time. Imagine a world where you don't pour concrete or smelt steel to build a skyscraper. Like instead, you literally grow the structural beams in a vat of liquid protein, dieting their shape with holograms of light.
Yeah, or imagine looking at industrial pollution, like the thick, choking exhaust from a coal plant, and not seeing a climate disaster, but seeing the raw, perfectly packaged fuel for an engineered biological refinery.
Right. And imagine a hospital where a genetic disease isn't treated with, chemical poisons or invasive surgeries. It's simply patched. Exactly the same way you would download a software update to fix a glitch on your phone.
I mean, it sounds like the pitch for a science fiction novel, a highly optimistic one.
Totally.
But the most striking thing we're going to look at today is that the foundational mechanics for every single one of those concepts, they already exist. We aren't waiting on some magical leap in physics. The tools are sitting on the workbench right now.
And that is our mission for today's deep dive. We're exploring this truly massive, fascinating stack of speculative science and engineering essays written by independent researcher Zachary David Enfield in early 2026.
It's a really dense read, but so rewarding.
Oh, absolutely. The materials span 4 major papers outlining what he calls a unified architecture for the future of civilization. And the core premise, the fundamental paradigm shift that anchors all of this, is that biology is not just a messy, unpredictable natural process.
Right.
Biology is, without exaggeration, the most advanced, programmable, and information-dense technology on planet Earth.
And that framing is everything, Enfield argues that the distinction we draw between a machine, a computer, a living organism, and a piece of city infrastructure is, well, it's completely artificial. It's a semantic illusion.
Just totally made up by us.
Exactly. At their core, they are all just systems processing information and moving materials. The only difference is that biology happens to be a system that has had roughly 3.5 billion years of brutal, unforgiving trial and error to optimize its code.
Okay. Let's unpack this, because we have an incredible journey ahead of us today. We are going to start at the absolute microscopic level, the source code of human DNA.
The foundational stuff.
Yeah. And from there, we'll move into revolutionary new physical materials built from proteins that used to actually terrify us. We'll explore how lasers and light can literally 3D print these materials into functioning biological computers. And finally, we're going to scale all the way up to re-engineering the planet's entire agricultural and ecological network.
It's a progression that really demands we completely abandon our traditional industrial mindset. I mean, to treat biology as a technology, we first have to understand how to read, write, and execute its foundational code.
Right.
And that requires us to look at the human body and human illness through an entirely new lens.
Which brings us to Enfield's first major argument, right? Debugging the human machine.
Yeah.
If you've ever had a loved one get sick or dealt with a chronic illness yourself, you know that medicine historically relies on the language of war.
Oh, it is deeply ingrained in our culture.
Oh, completely. We talk about battling cancer. We talk about fighting off infection. We view the body as a battlefield, and the treatments really reflect that.
Yeah, they do.
Like chemotherapy is essentially carpet bombing the body with poison, just hoping the cancer dies a little bit faster than the healthy cells do. It's chaotic, it's destructive, and it implies this fundamental lack of precise control.
And Enfield insists we have to throw that entire lexicon out the window. A genetic condition or a misbehaving cell is not a monster attacking you. It is a failure of information.
A failure of information.
Exactly. It's corrupted code running on the living cell's computational substrate. I mean, if you view a tumor as an invading army, your instinct is to drop a bomb on it. But if you view a tumor as a corrupted loop in a software program, your instinct changes entirely.
Yeah, you don't bomb a software bug, you debug it.
Precisely. You find the error in the sequence, the specific line of bad code, and you correct it at the source. But the monumental challenge in biology has always been delivery.
Right, getting it in there.
Yeah. How do you get your new corrected line of code inside the nucleus of a living cell without destroying the cell in the process? You need a courier, like a microscopic vehicle that knows exactly how to navigate the cellular membrane.
And nature already built the perfect courier. For billions of years, there has been an entity whose sole evolutionary purpose is to cross cell membranes, evade immune detection, and deliver a payload of genetic information into a host. The virus.
The virus it's the ultimate masterpiece of nanoscale engineering. We're so conditioned to view viruses exclusively as pathogens, like as the ultimate enemy.
Right, because they make us sick.
Exactly. But a virus, stripped of its context, is just a mechanism. It's a mathematical shape, usually an icosahedron, which is a 20-sided protein shell called a capsid, and it just surrounds a strand of nucleic acid.
I was reading this section and I couldn't stop thinking about a USB flash drive.
That's a good way to look at it.
Right, like if you find a random USB drive on the sidewalk, the danger isn't the plastic casing. The danger isn't the metal connector that plugs into your laptop. The danger is the malware stored on the drive.
Right.
But if you take that exact same USB drive, plug it into an isolated terminal, format it, wipe the malware, and load it with a critical software patch that your computer desperately needs, that exact same piece of hardware goes from being a catastrophic threat to a life-saving tool.
That is a brilliant way to conceptualize it. What makes a virus pathological isn't its physical architecture, it's the payload inside. The mechanism of entry is entirely neutral.
Okay, so how do we use that?
Well, when human engineers take a virus, commonly things like adeno-associated viruses or AAVs and lentiviruses, they literally open them up and strip out the harmful viral instructions. The virus can no longer replicate. It can no longer cause disease. It is just an empty shell.
And then we pack our own code inside.
Exactly. We replace the malware with therapeutic instructions. And once we do that, the virus ceases to be an enemy. It becomes what Enfield terms a nanite.
A nanite, wow.
Yeah. It's a programmable biological machine built for healing. And just think about the sheer efficiency of this. Human engineers don't have to spend decades trying to invent a synthetic, nanoscale drug delivery vehicle from scratch. We don't have to figure out how to bypass the lipid bilayer of a cell. We just inherit 3 billion years of evolutionary optimization for free.
Okay, so we have the USB drive. We've wiped the malware. What is the software patch we are actually putting inside these viral nanites? The payload is a molecular text editor, specifically CRISPR-Cas9.
And we should really break down what that actually means mechanically, because it gets thrown around a lot as a buzzword.
Yeah, it definitely does.
CRISPR isn't a synthetic invention. It's originally a bacterial immune mechanism. Bacteria actually use it to chop up the DNA of viruses that attack them. But scientists realized they could repurpose the system into a highly programmable molecular scalpel.
But how does the scalpel know where to cut, though? I mean, the human genome has 3 billion base pairs. If you cut in the wrong spot, you don't cure a disease, you just cause a new one.
That's where the guide RNA comes in. The payload we put inside the viral nanite includes 2 vital components. First, the Cas9 protein, which acts as the physical scissors. And second, a custom-designed strand of guide RNA.
Okay, so it has a guide.
Right. This guide RNA is like a GPS coordinate. It flips through the nucleus, scanning the billions of letters of DNA until it finds the exact 20-letter sequence that it perfectly matches.
So it physically binds to the corrupted code.
Yes, it binds to the target, which locks the Cas9 protein into place. The Cas9 protein then undergoes a physical shape change and cleaves the DNA double helix right at that exact location. It physically breaks the corrupted gene.
And then what happens?
Well, from there, the cell realizes its DNA is broken and basically panics. It activates its own natural repair machinery. We can either let the cell glue the broken ends back together clumsily, which effectively disables the faulty gene, or we can provide a corrected template for the cell to use as a blueprint during the repair.
That is wild. And you know, if you're hearing this, you might be thinking, this is still in a lab somewhere, like something we'll see in 20 years. But Enfield explicitly points to the FDA approval of Casgevy in December of 2023. This isn't theory. This is history.
Yeah, it's already here.
Casgevy is a therapy for sickle cell disease, which is a devastating and incredibly painful genetic disorder.
It really is. Sickle cell is caused by a single typo in the DNA sequence for hemoglobin, which is the protein that carries oxygen in the blood. That single error causes the red blood cells to fold into rigid sickle shapes that get stuck in blood vessels.
So how does Casgevy fix it? Because it doesn't actually fix the broken adult hemoglobin gene, does it? It does something much cleverer.
It uses a fascinating biological workaround. So as fetuses in the womb, humans produce a different kind of hemoglobin called fetal hemoglobin, and it's extremely efficient at grabbing oxygen.
Okay.
Shortly after birth, a specific gene called BCL1A acts as an off switch to shut down the fetal hemoglobin production, and the body switches to adult hemoglobin.
But in sickle cell patients, the adult hemoglobin is broken.
Exactly. So Casgevy uses CRISPR-Cas9 to target that BCL11A off switch and physically break it.
Oh, wow.
Yeah. By breaking the off switch, the body reverts to its default programming and starts producing healthy, perfectly functional fetal hemoglobin again. It bypasses the corrupted adult code entirely.
And the clinical results are just staggering. In the trials, 29 out of 31 patients were completely free of severe pain crises for at least a year following a single treatment. I mean, they took patients' own stem cells, edited them outside the body, and put them back in.
It's incredible.
That isn't symptom management. That isn't a daily pill. That is literally debugging the source code to effect a permanent cure.
And while editing cells outside the body ex vivo is an incredible milestone, the trajectory Enfield focuses on is systemic in vivo delivery, editing happening directly inside the living, breathing human body.
Right.
He points to therapies like NTLA-2001 for a condition called hereditary amyloidosis. That's where the liver produces a toxic, misfolded protein that destroys the nerves and heart.
So how does that work in the body?
They encase the CRISPR machinery inside lipid nanoparticles, which are just tiny fat bubbles, and infuse it directly into the patient's bloodstream. The nanoparticles naturally accumulate in the liver, enter the liver cells, and the CRISPR machinery physically cuts the gene responsible for the toxic protein.
And it works.
Clinical data showed a greater than 90% reduction in those toxic proteins circulating in the blood.
From a single infusion?
A single infusion. Or look at CTX310, which Enfield highlights from a 2025 trial. This targets a gene called ANGPTL3 in liver cells. By permanently switching off this one gene, they cut LDL cholesterol by 50% in patients with severe resistant lipid disorders.
Imagine going to the doctor with a family history of fatal heart attacks, getting one single IV infusion, and your cholesterol is permanently lowered by 50% for the rest of your life.
It's life-changing.
It fundamentally alters the human timeline. But here is where Enfield takes a massive leap. If we can patch broken human code to fix diseases, the natural, inevitable next question is, what happens when we start writing new code?
Right.
What happens when we stop just fixing what's broken and start looking at the massive open source library of all life on Earth to borrow upgrades?
And this is the critical transition from traditional genetic engineering to what we now call synthetic biology. In the 90s and early 2000s, genetic engineering was essentially cut and paste. It was blunt.
Super messy.
Very. You would take raw, messy DNA from a jellyfish that made it glow and transplant that exact raw sequence into a mouse, just hoping the mouse's cellular machinery could read it.
It's like trying to run an ancient Mac software program on a modern Windows computer.
Yeah.
Sometimes you can force it to work, but it's glitchy and totally inefficient.
Exactly. Synthetic biology, on the other hand, says we don't need to copy the literal DNA sequence from the jellyfish. We just need to understand the functional mechanism of the trait. Right? The physics and chemistry of how the trait works. Once we understand that, we can open up a digital text editor and write a bespoke, entirely new DNA sequence from scratch. A sequence that codes for that exact same trait, but is optimized specifically for the human cellular environment.
Wow.
Enfield calls this functional translation. You aren't mixing species. You are bypassing the species barrier entirely by writing native code.
And the examples he pulls from the Tree of Life to functionally translate are unbelievable. Let's start with the tardigrade, the water bear.
Oh, I love tardigrades.
They're amazing. These are microscopic creatures that are famous for being virtually indestructible. You can freeze them to near absolute zero, boil them, crush them. But the most relevant superpower is that they can survive 3,000 times the lethal human dose of ionizing radiation. They can survive the raw vacuum and radiation of outer space.
That's hard to even comprehend.
How is a biological organism capable of surviving that kind of physics?
Well, to understand the tardigrade's survival, we have to understand what radiation actually does to a living cell. When high-energy gamma rays or X-rays pass through a human cell, they hit water molecules and literally blast them apart. This creates what are called hydroxyl radicals.
Okay, what are those?
They're highly unstable reactive oxygen species that act like microscopic shrapnel.
So it's not the radiation itself that directly kills you, it's the shrapnel the radiation creates out of your own water.
Most of the time, yes. That shrapnel ricochets around the nucleus and physically severs the double helix of your DNA. If the DNA sustains too many of these double-strand breaks, the cell's repair machinery is overwhelmed, the code is shredded, and the cell undergoes apoptosis. It dies. That is radiation sickness.
So how does the tardigrade stop the shrapnel?
The tardigrade evolved a highly specialized, unique protein called DSUP, which literally stands for damage suppressor. It is a highly unstructured, flexible protein. And what it does is physically bind to the nucleosomes. Those are the spools that the tardigrade's DNA is wrapped around. It drapes itself over the chromatin like an impenetrable microscopic blanket.
Just a physical shield.
Physical electrostatic shield. When the hydroxyl radicals approach the DNA, the DSUP protein intercepts them and neutralizes them before they can sever the double helix.
And Enfield notes that we have functionally translated this. MIT researchers actually took the code for this tardigrade protein, synthesized it into an mRNA sequence, and delivered it into human cells using lipid nanoparticles. And the human cells suddenly gained acute radioprotection.
It's brilliant.
The radiation shrapnel couldn't shred the human DNA because the synthetic tardigrade blanket was protecting it.
The implications for medical and space applications are profound. Because it's delivered via mRNA, the effect is temporary. The mRNA translates the protective protein, the cell uses it, and then the mRNA naturally degrades within hours. There is no permanent modification to your genome.
Think about what that means. You could give a localized infusion of DSUP mRNA to a cancer patient's healthy tissue right before they undergo intense radiation therapy. The tumor, which didn't get the DSUP, is destroyed by the radiation, but the surrounding healthy organs are completely shielded.
Exactly.
Or an astronaut on a Mars mission gets a solar flare warning alert. They inject a syringe of synthetic DSUP, their cells deploy the radiation shields for the next 24 hours, and they survive the solar storm unharmed.
It is protection on demand. But Enfield goes far beyond extremophile shielding. He looks at active structural regeneration. He looks at salamanders, specifically the axolotl.
Oh, this part was wild.
It's an amphibian that possesses the holy grail of regenerative biology, the ability to regrow entire, perfectly structured, amputated limbs.
And this isn't just a lizard growing back a tiny cartilage tail. If you cut off an axolotl's leg, it grows back the bone, the muscle, the complex vascular system, the nerves, and the skin. Perfectly.
Perfectly.
How does the stump of a severed arm know how to grow a hand instead of just growing a chaotic tumor of meat?
It's all about cellular memory and morphogen gradients. When an axolotl loses a limb, the cells at the site of the amputation undergo an incredible process called dedifferentiation. A specialized muscle cell or a bone cell basically forgets its adult identity and reverts back into a flexible stem cell-like state. These reverted cells gather at the wound site to form a mass called a blastema.
And the blastema is basically the raw clay.
The raw clay, yes, but the clay contains architectural blueprints. The cells release chemical signaling molecules in specific concentrations gradients. A cell knows exactly where it is in 3D space based on the concentration of these chemicals.
Like a GPS system for the cells?
Right. If it senses a high concentration of signal A and a low concentration of signal B, its DNA executes the program to become a fingertip bone. The limb builds itself outward, layer by layer, driven purely by these chemical gradients.
But the craziest part of the research Enfield cites, and this is from a team at LSU, is that the genetic toolkit for this kind of complete blastema regeneration isn't some alien amphibian magic. Humans actually still have the code. We share the underlying genetic architecture.
That is the most staggering revelation. We aren't missing the genes required to form a blastema. They're just dormant.
Why?
Well, over millions of years of mammalian evolution, we prioritize rapid scarring over slow regeneration. If a primitive mammal got its leg bitten off by a predator, taking 3 months to slowly regrow a new leg wasn't a survival advantage. It would bleed out or die of infection in days.
Oh, that makes total sense.
So evolution strongly selected for rapid, thick fibrosis, scar tissue. The scar seals the wound instantly, but it physically blocks the formation of a blastema.
So the regeneration software is still on our hard drive. It's just been locked behind an evolutionary firewall.
Precisely. And if we connect this to the bigger picture of viral nanites, it means human limb or organ regeneration might not require introducing any foreign axolotl biology into our bodies at all.
Just a key.
Yes.
Yeah.
It might just require a viral nanite carrying synthetic transcription factors keys to unlock our own dormant genetic programs. We temporarily turn off the scarring mechanism and switch the blastema program back on.
It's awe-inspiring. Enfield even talks about looking at diving mammals like seals or whales to understand how their myoglobin is structurally optimized for extreme, long-term oxygen storage in muscle tissue. Or looking at the synthetic opsins in the retinas of owls, which could potentially be translated to give human retinas infrared vision capabilities.
The possibilities of a fully cataloged, functionally translated tree of life are literally boundless.
Okay, I have to step in and push back here. Because if you're listening to this, I know what you're thinking, because it's exactly what I'm thinking. I hear infrared vision and regrowing arms and radiation immunity, and part of me is amazed. But a massive part of me is terrified.
It's a very natural reaction.
We are talking about humanity deliberately redesigning its own species. Evolution has a speed limit for a reason. If we rip the brakes off and allow anyone with a bioreactor to rewrite human traits, what stops this from spiraling completely out of control? Don't we risk biological catastrophes?
That is a very real, very valid fear. Natural evolution is agonizingly slow, it's blind, and it's incredibly wasteful. But it has one massive advantage. It is safe, because systemic changes take hundreds of thousands of years to propagate.
Exactly.
What Enfield is outlining is programmable evolution. It is fast, it is directed, and it is intentional.
Yeah.
And you are entirely correct. If you introduce that kind of power without rigorous, inflexible architectural safeguards, the system will collapse.
So what do those safeguards actually look like? Because if someone edits their DNA to have enhanced muscle density, and then they pass that edit down to their children, and it turns out there's a fatal flaw in the code that we didn't foresee, we've corrupted the human gene pool permanently.
Which is exactly why Enfield insists on strict, absolute reversibility at the molecular level. For temporary applications, as we discussed with the tardigrade DSIP protein, you use mRNA. The protective effect literally cannot last longer than a few hours before the molecule naturally degrades. That is a passive kill switch.
But what about the permanent DNA edits? If I permanently rewrite my genome to activate the blastema regeneration, mRNA isn't going to cut it.
For permanent edits, the safeguard is a synthetic promoter activated by a small molecule.
Let's break down the mechanics of that.
Okay, so a promoter is a sequence of DNA that acts like the power switch for a gene. RNA polymerase, the enzyme that reads the gene, won't bind to the DNA unless the promoter invites it to.
So you replace the natural power switch with a synthetic one.
Yes. You write a bespoke promoter sequence that does not naturally turn on under any normal human biological conditions. The new trait, let's say the extreme oxygen storage in your muscles, is fundamentally off by default.
So how does it turn on?
The synthetic promoter will only recruit the polymerase and activate the gene if it physically binds to a highly specific, harmless synthetic chemical compound, a specific ligand.
So you literally have to take a specialized pill to turn your new DNA on.
Exactly. The genetic modification is totally inert until you introduce the chemical key. The trait requires continuous chemical dependency.
Wait, I'm still skeptical. What happens if you're on a hiking trip, you drop your pills in a river, and suddenly your synthetic biology turns off? Or worse, what if the DNA mutates? What if the synthetic promoter gets damaged and gets stuck in the amen position, ignoring the need for the pill entirely?
Those are the exact edge cases Enfield's architecture models. First, the dependency relies on steady-state pharmacokinetics, meaning missing a single pill doesn't instantly shock your system. The trait fades gradually as the chemical washes out of your bloodstream.
Okay, that makes sense.
Second, regarding mutation, the synthetic promoters are engineered with redundancy. They don't just require a single binding site, they require a complex multimeric assembly. The statistical probability of a random mutation perfectly replicating that exact geometric assembly to get stuck on is functionally zero.
So it should break instead.
Right. A random mutation is overwhelmingly likely to simply break the promoter entirely, returning the trait to the off state. Failure defaults to baseline human biology.
Okay, the mechanical safety features make sense, but there is an even bigger danger here, an ethical one. If this technology is priced the way cutting-edge medicine is priced today, I mean, early CRISPR therapies cost millions of dollars per dose, aren't we just building a permanent biological caste system? You'll have the physiologically enhanced, disease-immune wealthy class and the unenhanced baseline poor.
Enfield addresses this directly. He argues that functional translation and genetic augmentation cannot be allowed to exist as a luxury market. It must be classified as a global public infrastructure, exactly like clean water or basic sanitation.
Wow, that's a massive shift in how we view medicine.
It has to be. If it isn't, the societal divergence will be unbridgeable. The technology must uplift the baseline of humanity entirely, not fracture it.
It's a heavy thought, but it provides a perfect bridge. We've spent all this time talking about rewriting the DNA code, but DNA is just information. It's a blueprint. You can have the best blueprint in the world, but a blueprint doesn't build a house.
You need materials.
Exactly. The physical structures that DNA actually codes for are proteins. And this is where Enfield completely pivots his entire architectural framework into material science. What if one of biology's most infamous, historically feared protein structures is actually the greatest untapped physical building material of our time?
You are referring to the prion and amyloid flip.
I am. And I need you to explain this, because when anyone hears the word prion, they instantly think of mad cow disease. They think of incurable, terrifying, brain-destroying neurodegenerative conditions. How on earth does Enfield look at a prion and say, yes, let's use that to build airplane wings and skyscraper beams?
To understand the genius of this, we have to separate the disease from the underlying geometry. What is a prion, fundamentally? It's simply a protein that is folded into a very specific, incredibly stable three-dimensional shape. And it have one unique, defining superpower.
Which is?
When it bumps into another normally folded protein of the same type, it acts as a template. It physically forces the normal protein to unfold and refold into the prion's exact shape.
It's like a microscopic zombie. One prion touches a healthy protein and suddenly you have two prions. They touch two more and you have four.
Exactly. It is a runaway, self-replicating, templating cascade. In the delicate, tightly regulated environment of the human brain, this cascade is disastrous. The prions aggregate into massive, insoluble plaques that physically tear neurons apart. That is mad cow disease. That is Creutzfeldt-Jakob disease.
Terrifying stuff.
But if we step out of the biology lab and walk into a material science lab, a self-assembling, totally insoluble, geometrically flawless, indestructible protein cascade isn't a disease, it's a miracle material.
Because the underlying structure of a prion is something called a cross-beta architecture. Explain what that is physically.
Well, proteins fold based on thermodynamics. They want to find their lowest energy state. The cross-beta architecture is essentially the absolute bottom of the folding funnel. The protein strands line up side by side, perfectly flat, and bond together using a massive, dense network of hydrogen bonds. They stack like an infinite deck of microscopic playing cards, forming long, continuous amyloid fibrils.
And structurally, because of all those hydrogen bonds, this material is unbelievable, right?
It is a highly tunable nanocomposite. When scientists engineer these polymeric amyloid fibers in a vat using safe functional sequences, totally divorced from any disease-causing genes, mechanical specifications are staggering. Let's talk about toughness, which is the amount of energy a material can absorb before it shatters.
Okay.
Engineered amyloid composites can achieve a toughness of 160 megajoules per cubic meter.
Wait, for context. Spider silk, specifically dragline silk, has always been the gold standard for biological toughness. It's the cliche everyone uses, like stronger than steel.
Right.
Does this beat spider silk?
It surpasses natural spider dragline silk, and it does this while being incredibly lightweight. Enfield notes the density of an amyloid composite is roughly 1/8 the density of structural steel.
So it's stronger than spider silk and 8 times lighter than steel. But what about chemical stability? Because proteins usually denature. Like, if you crack an egg into a hot pan, the clear protein instantly turns white and solidifies because the heat destroys its structure. If we build a bridge out of proteins, won't it melt in the sun?
Normal proteins denature easily. Cross-beta amyloids do not. Their chemical stability is almost unparalleled in organic chemistry. Fibrils formed from these engineered sequences can survive pure, concentrated formic acid.
Wait, pure acid?
Yeah. They can survive boiling lye. Conditions that would instantly dissolve a human body don't even dent the fiber. Enfield even cites studies showing they survive gamma ray irradiation at doses that would completely destroy a silicon-based computer logic board.
Okay, but if it's that rigid, how do we control it? If I need a rigid beam for a building, I want it tough. But if I need a flexible, dynamic gel for a medical implant inside a human joint, I can't use something that rigid.
That is where the hydrogen bond density design dial, or HBD, comes into play. Because we are designing the DNA sequence that makes the protein, we control exactly which amino acids are present. The mechanical rigidity of the fiber is directly proportional to the density of the hydrogen bonds locking the cross-beta sheets together.
So it's literally a dial we can turn in the code.
Exactly. It's the ultimate control knob. If you want a material that acts like rigid structural steel for an aerospace application, you write a sequence with tightly packed amino acids that maximize the hydrogen bond density. The sheets lock together like superglue.
And for the gel.
If you want a dynamic, fragile, water-rich hydrogel for a tissue scaffold, you swap out a few amino acids to intentionally disrupt the packing, decreasing the hydrogen bond density. It is the exact same base material, grown using the exact same process, just sequenced slightly differently to yield entirely different physics.
And the manufacturing process is the part that really broke my brain. Enfield calls it seeding, because if you want to make steel, you have to rip iron ore out of the earth, put it in a massive blast furnace, burn gigatons of coal, smelt it, cast it, and cool it. It is incredibly energy intensive and highly polluting. How do you manufacture 100 tons of amyloid composite? I'm picturing a giant brewery.
That is precisely what it is. You don't build this material in a factory, you brew it in a bioreactor. Imagine a massive stainless steel vat filled with liquid protein monomer. Just harmless, disconnected, folded proteins floating in water.
The raw material.
Yes. And then you introduce the seed. You take a microscopic fragment of a correctly folded amyloid fibril, and you drop it into the vat.
This reminds me of supercooled water. Like if you have a bottle of incredibly pure water and you freeze it below 32 degrees, it sometimes stays liquid because the ice has no impurities to grab onto to start forming crystals.
Right.
But the second you drop a single tiny ice crystal into that supercooled bottle, the entire bottle instantly freezes solid right before your eyes. The crystal acts as a template for the whole liquid.
That is a perfect physical analogy. The seed acts as the nucleation point. It bumps into a monomer floating in the liquid and forces it to fold and lock onto the end of the fiber. Then it grabs the next one and the next. The fiber grows in length. But the bioreactor is constantly agitating the liquid, stirring it. The mechanical shear force occasionally snaps the long-growing fiber in half. And now you have 2 seeds, which continue to grow and snap, creating 4 seeds, then 8, 16. It is exponential self-replication. You drop 1 seed into a 10,000-gallon vat of protein, and within hours, the entire vat has crystallized into dense, interwoven amyloid fibers. It scales at near zero marginal cost. You don't need heat. You don't need blast furnaces. You just need a protein-rich liquid medium and a motor to stir it.
If we connect this to the bigger picture, we're talking about replacing the entire global steel and concrete industry, which is responsible for a massive percentage of global carbon emissions, with room temperature vats of water and protein. But where does the raw protein come from? We can't exactly drain the world's agriculture to make building supplies.
We don't have to. The base feedstock for that protein can be grown sustainably from engineered microalgae. Algae requires nothing but sunlight, water, and CO2. You can grow the feedstock in barren deserts using salt water.
And because it's fundamentally a biological material, Enfield points out that it can be self-repairing. Let's talk about the biological armor concept.
Right. Imagine a structural panel on the hull of a deep-sea submarine or an aircraft fuselage made of this dense amyloid composite. Embedded within the rigid fiber matrix are microscopic pockets of liquid protein monomer. Under massive stress, let's say the panel takes an impact and a microcrack forms in the structure.
In a steel hull, that crack slowly propagates until the whole thing fails.
But in the amyloid hull, the crack physically tears open those microscopic pockets of liquid monomer. The liquid rushes into the crack. But what happens when liquid monomer touches the raw, broken edge of an amyloid fiber?
The seeding cascade restarts.
Exactly. The broken edges act as fresh seeds. The liquid monomer instantly begins to fold and lock into place, bridging the gap. The crack literally regrows itself, solidifying and healing the structural damage in real time. It is armor that senses damage and repairs it autonomously.
It's biological armor that heals. That is genuinely wild. But Enfield doesn't stop at making strong, self-healing panels. He pushes this cross-beta material straight into the realm of advanced electronics. These fibers aren't just strong, they can actively conduct energy and information. We are looking at the foundational wiring for biological computers.
This is where materials science seamlessly merges with nanoelectronics. These amyloid fibers can act as functional nanowires. The most basic way is to metallize them. You can chemically treat the outside of the protein fiber so that silver or gold atoms bind to it. The biological fiber just acts as a perfectly straight, self-assembling scaffold for a metallic wire that is only a few nanometers thick.
But Enfield highlights something much more profound. They don't even need the gold. The proteins themselves can conduct electrons intrinsically. How does a wet protein conduct electricity? It's not a metal.
It relies on a quantum mechanical phenomenon called tyrosine hopping. Within the amino acid sequence of the amyloid core, engineers can place a high concentration of an amino acid called tyrosine. Tyrosine has an aromatic ring, a hexagonal shape made of carbon atoms. In chemistry, these rings have what we call delocalized electron clouds.
Okay, what does that mean?
The electrons aren't locked to one specific atom. They hover above and below the ring in a pi orbital.
Okay, so you have these hovering electron clouds.
Because the cross-beta architecture packs everything so densely, the tyrosine rings from one layer are stacked perfectly on top of the tyrosine rings in the next layer. The distance between them is sub-nanometer. Their electron clouds actually overlap. This is called pi-p stacking. It creates a continuous overlapping pathway straight down the center of the fiber.
I'm picturing a river. If you throw a handful of stepping stones into a river randomly, you can't cross. The gaps are too wide. But if you arrange the stepping stones perfectly in a line, exactly 1 foot apart, you can easily hop from one to the next all the way across.
A perfect analogy. The electrons are the jumper, and the overlapping tyrosine clouds are the perfectly spaced stepping stones. It allows electrons to literally hop along the length of the fiber over micrometer distances. You don't need copper, you don't need silicon. The biological sequence itself dictates the conductivity.
And the fibers don't just conduct electricity, they can generate it. Enfield notes these fibers are highly piezoelectric. Let's break down what piezoelectricity actually is, because it's a critical component here.
Piezoelectricity is a property of certain crystalline structures, where mechanical stress is converted directly into electrical voltage. If you physically squeeze or compress the crystal lattice, it forces the positive and negative charges inside the material to shift to opposite sides, creating a voltage potential.
And these amyloid fibers do that.
Incredibly well. If you apply mechanical pressure to them, even ambient acoustic pressure like sound waves or ultrasound, they generate electricity. Enfield points out they can generate up to 1 volt of open circuit voltage just from ambient vibrations.
Think about the medical applications of that. You could have a biosensor implanted inside your heart to monitor arrhythmias, and it doesn't need a battery. It doesn't need wires passing through your skin. It powers itself entirely by harvesting the piezoelectric energy generated by the acoustic thump of your own heartbeat.
Or consider active therapeutics. One incredible study Enfield cites involved injecting these piezoelectric fibers directly into a deep tissue tumor. From the outside of the body, doctors applied targeted ultrasound waves. The sound waves penetrate the tissue and hit the fibers.
And they generate a charge.
Right. The fibers vibrate, and the piezoelectric effect generates localized electrical charges right inside the tumor. This electrical charge splits water molecules to create those reactive oxygen species, ROS, that we talked about earlier. The localized shrapnel selectively destroys the cancer cells from the inside out. It's wireless, battery-free cancer therapy driven by sound.
So we have biological wires that can power themselves, heal themselves, and conduct data. What happens when we try to build a computer out of them? This brings us to one of the most mind-bending concepts in the entire stack of essays, Enfield's quaternary quantum processor.
This is the synthesis of everything we've discussed. We take DNA, which we already established is the densest information storage medium in the universe, and we use it as the hard drive. Think about a traditional silicon computer. It uses binary logic, ones and zeros, 2 states.
Right, on or off.
But DNA naturally has 4 states. The 4 nucleotide bases, adenine, cytosine, thymine, and guanine, A, C, T, G. That makes it a quaternary system, not a binary one. The information density increases exponentially. You combine that immense quaternary data storage of the DNA with the Prion nanowires we just discussed, which act as the logic gates in the physical circuitry connecting the DNA clusters.
But a processor needs a clock speed. A traditional CPU has a quartz crystal that vibrates billions of times a second to synchronize the calculations. What makes the DNA Prion processor tick? How do you actually read and write the data quickly? Enfield says it's driven by terahertz radiation, pulses of terahertz light.
Yes. The terahertz band sits between microwave and infrared light on the electromagnetic spectrum. What physicists discovered is that terahertz pulses resonate perfectly with the specific frequencies of the hydrogen bonds that hold the 2 strands of the DNA double helix together.
We resonate, like a singer breaking a wine glass with their voice.
Exactly like that. The singer hits the exact resonant frequency of the glass vibrates wildly, and the physical bonds shatter. But with the terahertz light, we aren't shattering the DNA. We apply a highly calibrated, ultra-fast pulse. The resonance causes the hydrogen bonds holding the AT and CG pairs to temporarily vibrate apart.
So it unzips it.
It unzips the double helix, exposing the data so it can be read or modified by a biological logic gate. And then, milliseconds later, the pulse stops and the helix instantly snaps back together. It rezips.
How does using light to vibrate chemical bonds apart not just cook the DNA? I mean, if I put DNA in a microwave, the radiation heats the water and boils the protein.
Because terahertz radiation is non-ionizing, and the pulses are on the picosecond scale, trillionths of a second. The energy is delivered purely as mechanical resonance to the hydrogen bonds, not as thermal heat to the surrounding water. You get incredibly rapid read and write speeds without generating the thermal damage that would normally denature a biological molecule.
Okay, I have to stop and ask the critical question, because if you're hearing this, you're probably trying to visualize this machine and struggling. I certainly am. We have DNA storing data. We have prion fibers acting as wires. We have terahertz light zipping it all together. How does this not just turn into a puddle of organic mush?
That's the challenge.
Yeah. How do these wildly different components physically interface with each other? We've tried biological computers before, attaching rat neurons to silicon chips, and they always fail.
It's the most important engineering question you could ask, and the answer reveals the true brilliance of Enfield's architecture. It comes down to scale matching. In traditional hybrid technology, trying to interface silicon chips with biological tissue, they clash because of a massive dimensional mismatch.
Right. A silicon transistor, even a microscopic one, is a fundamentally different physical texture and scale than a wet biological cell membrane. It's like trying to connect a massive fire hose to a microscopic capillary vein. It doesn't fit, it leaks, and it breaks.
Exactly. Even our most advanced silicon fabrication, getting down to 3 or 2 nanometers, is incredibly brittle and runs into quantum tunneling issues. But in Enfield's computational soup, look at the physical dimensions. The Prion nanowires are intrinsically about 10 nanometers wide. That perfectly, seamlessly matches the molecular scale of the DNA origami structures.
Ah, so there is no interface mismatch.
None. The storage medium, the logic gates, and the wiring exist at the exact same physical nanometer scale. They are made of the exact same families of amino acids and nucleic acids. They float in the same aqueous environment. It creates a seamless computational architecture. The structural dimensions natively align because they are all born from the same biological toolkit.
That is deeply satisfying from an engineering perspective. But there is one glaring missing piece to this entire puzzle. We have these amyloid fibers that we know grow exponentially in a vat. They self-replicate like crazy when we drop a seed in.
Yeah.
But how do we actually force them to form the precise, complex macro shapes we need?
You need to control them.
Exactly. If I want to manufacture a biological circuit board or a replacement human femur bone or the wing of an aircraft, I can't just rely on a chaotic, random tangle of fibers growing in a bioreactor. How do we draw the blueprint into the liquid?
That is the ultimate manufacturing problem, and it is solved by a process Enfield calls Prion optogenetic lithography.
Writing matter with light.
Yes, it sounds almost magical, but it is the rigorous convergence of 3 completely separate scientific pillars. First, the Prion cascade, which we know gives us the exponential amplification and material growth. Second, optogenetics, which gives us precise, light-responsive control over protein behavior. And third, photolithography, which is the spatial patterning technique we currently use to print computer chips using light.
Let's break down the optogenetics part because that's the key. Optogenetics is usually used in neuroscience to make neurons fire when you shine a light on them. How are we applying it to building materials? Enfield mentions fusing light-sensitive domains, specifically proteins called Cry2 or LOV2, directly to our prion-forming sequences. What does a light-sensitive domain actually do mechanically? How does a protein see light?
Well, it doesn't see it. It physically absorbs the energy of the photon. Inside the LOV2 protein domain, there is a specific molecule called a chromophore. When a photon of a very specific wavelength of blue light hits that chromophore, the energy causes a double carbon bond inside the molecule to briefly snap into a single bond. This allows the molecule to physically rotate and twist.
A physical shape change driven by light.
Exactly, a conformational change. We take that LOV2 domain and we genetically fuse it over the top of our prion sequence like a cap. In the dark, the LOV2 domain holds the prion protein in a tightly locked folded state. The prion sequence is hidden. The proteins remain as completely harmless, disconnected liquid monomers, just floating peacefully in the vat. They cannot fold, they cannot aggregate.
But when the light hits them,
The moment they are illuminated with that specific wavelength of blue laser light, the chromophore absorbs the photon, the domain twists, and it pulls the cap off. It exposes the sticky prion sequence. The protein unlocks and immediately begins to self-assemble into fibers.
and crucially, it only self-assembles exactly where the light touches it. The liquid in the dark stays liquid. The liquid hit by the laser turns into solid, steel-like fiber. Enfield highlights the phobite system, which refines this even further. It acts as precise light on and light off switches. You use blue light to make it solidify, and maybe a pulse of red light to force it to unlock and dissolve back into liquid.
It gives you absolute spatial and temporal control over a self-replicating material. You can literally project a holographic pattern of light into the center of a vat of liquid, and the solid material will only crystallize within the exact three-dimensional boundaries of that hologram.
The applications for this are they reshape everything. Enfield talks about protein lattice printing for food. This completely bypasses the current lab-grown meat bottleneck.
Oh yeah, the texture issue.
Right. Right now, lab-grown meat is just a mush of cells because they can't replicate the complex vascular and fibrous structure of a real steak. But with this, you don't grow cells. You just project a high-resolution hologram of a muscle's microarchitecture into a vat of nutritional proteins. The light writes the aligned fiber bundles, the chew, the texture. You print a perfect, highly structured steak without ever involving an animal.
Or look at trauma medicine. Imagine a patient comes in with a shattered femur. You don't try to screw metal plates into the remaining bone. You inject these liquid-locked monomer constructs directly into the wound site. Then, the surgeon uses a fiber optic laser to project a 3D regenerative scaffold hologram directly inside the living tissue.
Oh, wow.
The light passes through the flesh, hits the liquid pool, and builds a solid, weight-bearing, porous amyloid support structure exactly where it's needed. The patient's own bone cells then migrate into that scaffold and replace it with natural bone over time.
So to bring this vividly to life for you listening, think about how a normal 3D printer works today. It has a mechanical arm, a heated nozzle, and it slowly extrudes melted plastic, layer by tedious layer. It has moving parts, it's slow, and the resolution is limited by the physical size of the nozzle. What Enfield is describing is a 3D printer with absolutely zero moving parts.
Not at all.
You have a quiet, still vat of liquid protein. You shine a complex 3D hologram of light into the center of the liquid, and a solid, perfectly structured, indestructible object rapidly crystallizes out of the liquid in real time, exactly matching the light. It's almost like a biological replicator from Star Trek.
It's a beautiful image, but from an engineering perspective, it raises a vital question regarding resolution. How detailed can this holographic object actually be? And the answer is the profound magic of the biological cascade.
Because light has a limit, right? The diffraction limit.
Exactly. We can focus lasers down to the diffraction limit, roughly half the wavelength of the light, which puts us in the scale of hundreds of nanometers. But we need structures that are accurate down to the single nanometer. How do we cross that gap? The laser light dictates the starting point, the initial nucleation event. But the moment the light unlocks that first protein, the biological prion cascade takes over.
The seed is planted. The seed is planted by the light. And the biological cascade mathematically amplifies that tiny starting structure into the macro world with atomic precision. The light writes the initial geometry, and the thermodynamic inevitability of the prion folding builds it out perfectly. It is the ultimate bridge between software, the digital pattern of the light, and physical reality, the solid atomic protein structure.
It is genuinely breathtaking. We've re-engineered the human cell with viral couriers. We've synthesized extremophile biology to augment our resilience. We've re-engineered the physical materials of our world and figured out how to write biological matter with lasers. But Enfield doesn't stop at the microscale.
No, he scales it all the way up.
In his final essay, he takes one massive step backward. He applies this exact same programmable biological logic to the macro world, to the entire planet's industrial, agricultural, and ecological infrastructure.
He calls this final phase healing the macro. And it starts by tearing down what he calls the linear fallacy of modern human civilization.
The linear fallacy. This is the idea that our global economy essentially operates as a massive one-way street. We extract raw resources from the earth, we burn them or manufacture them into products, and then we discard the toxic waste at the end of the line. It's a straight line from extraction to pollution.
And Enfield makes a profound unifying connection here. A genetic disease in a human body, an ecological collapse on a planetary scale are the exact same mechanical problem, just manifesting at vastly different scales. In both cases, you are looking at a system that is failing to process information and materials correctly. Both are feedback loops that have been severed.
In nature, there is no such thing as waste. The concept of waste is a uniquely human industrial invention. In a forest, the dead leaves that fall from a tree aren't waste. They are the exact required carbon and nitrogen feedstock for the fungal network in the soil, which in turn provides nutrients back to the tree's roots. The loop is closed.
Precisely. And the solution to our broken industrial loops is to close them, relying on a philosophy Enfield calls planetary citizenship. This means we must stop treating biological systems as passive resources to be mined or paved over, and start treating them as active, reciprocal, programmable infrastructure.
Let's look at how he applies this to one of the most stubborn problems we have: heavy industry and coal power plants. A coal plant's exhaust smokestack is packed with highly concentrated CO2 and heavy metals. Currently, we either vent it into the atmosphere, driving climate change, or we try to capture it using massive mechanical fans and pump it deep underground, which takes almost as much energy as the plant produces in the first place.
It's totally inefficient.
Yeah, Enfield completely sidesteps that with bio-CCS, bioenergy with carbon capture and storage, or what he simply calls algae scrubbers.
The elegance of bio-CCS is that it views the exhaust not as a poison, but as a rich, misplaced nutrient. You take the concentrated CO2 exhaust straight from the coal plant or an ethanol refinery, and instead of venting it up, you pipe it directly into a massive array of adjacent, shallow water bioreactors. These bioreactors are seeded with specific strains of engineered microalgae.
And CO2 is plant food.
It's the primary building block of plant life. Because you are piping in artificially high concentrations of CO2, it forces the algae into a state of explosive, hyperaccelerated growth. The algae literally eats the pollution in real time, scrubbing the exhaust clean before it ever reaches the atmosphere.
But then you have a massive pond full of algae. What do you do with it?
The specific microalgae strains chosen have been metabolically engineered to have extremely high intracellular lipid content. Lipids are fats, oils. Once the algae has absorbed the carbon, you harvest the biomass, dry it, and physically press it to extract the lipids. That extracted oil is a rich, completely sustainable, energy-dense biodiesel. You can put it straight into an airplane or a tractor. You have taken the toxic waste from a 20th century coal plant and used a biological compiler to transform it into the raw, clean fuel for the next cycle.
The waste becomes the fuel. He applies this exact same circular logic to agriculture with what he calls the duckweed loop. Modern farming is heavily reliant on massive applications of synthetic nitrogen and phosphorus fertilizers. A large percentage of that fertilizer doesn't get absorbed by the crops. It washes off the fields when it rains, flows into local rivers, and eventually empties into the ocean, causing massive, toxic algal blooms and hypoxic dead zones. The Gulf of Mexico dead zone is the classic example.
Again, Enfield diagnoses this not as an unsolvable environmental tragedy, but simply as misplaced fertility. The nitrogen and phosphorus are doing exactly what they are designed to do: fuel rapid plant growth. They were just doing it in the ocean where they suffocate fish instead of on the farm.
So the solution isn't to ban fertilizer and let crops fail. The solution is to intercept the misplaced fertility before it leaves the system. You build shallow, winding canals of duckweed along the drainage edges of every agricultural field. Duckweed is a tiny, floating aquatic plant, and it is arguably the fastest-growing vascular plant on planet Earth. Under ideal conditions, it can double its entire body mass in a matter of 24 to 48 hours.
And its primary fuel is nitrogen and phosphorus. As the fertilizer runoff washes off the field, it enters the duckweed channels. The duckweed aggressively intercepts and absorbs the nutrients, multiplying exponentially. The water that exits the far end of the channel and flows into the river is completely stripped of excess nutrients. It prevents the aquatic dead zones entirely.
But the genius is the second half of the loop. The duckweed doesn't just trap the waste. The duckweed becomes a hyper-condensed packet of exactly the nutrients the farm needs. You mechanically skim the duckweed off the channels, compost it, or run it through an anaerobic digester, and you have just manufactured a powerful, rich biofertilizer.
And you just put it back.
Yeah, you take that compost and put it right back onto the crop fields. The fertilizer that tried to wash away was caught, repackaged by a biological nanite, the duckweed, and returned to the soil.
It creates a perfectly closed, localized nutrient cycle.
I want to dive deep into one specific example Enfield uses, because it perfectly encapsulates how our historical obsession with linear mechanical efficiency actually breaks ecosystems. Let's talk about the Ogalla aquifer crisis. The Ogalla is a massive, ancient, underground reservoir of water that spans beneath the Great Plains of the United States. It's the water source that feeds a huge portion of American agriculture, and right now it is rapidly running dry. Enfield points out what he calls the efficiency paradox of modern irrigation.
This is a classic tragic example of treating a complex, multivariable biological system as a simple linear math problem. Historically, farmers in Nebraska and Kansas used gravity ditches. They dug earthen canals and flooded the furrows between the crops. From a purely mechanical standpoint, it was disastrously inefficient. The water seeped deep into the dirt where the roots couldn't reach. It pooled in low spots, and massive amounts of it evaporated into the hot summer air.
Engineers looked at that and said, we're losing over half our water. We need to optimize. So we invented center pivot irrigation. We've all seen them from an airplane window, those giant green circles in the middle of the desert. It's a massive mechanical sprinkler arm on wheels, slowly rotating, applying highly pressurized water directly to the base of the plant with 98% efficiency. Not a drop wasted. Problem solved, right?
Completely wrong. The brutal paradox is that making the irrigation delivery 98% efficient actually caused the ancient aquifer beneath them to drain faster.
How is that physically possible? I mean, if you're using less water per plant, the reservoir should last longer.
Because the engineers failed to map the macrobiological loop. Those leaky, terribly inefficient old gravity ditches were actually performing two vital, invisible ecosystem services that we completely relied on but didn't understand until we removed them. First, the wasted water seeping deep into the dirt was actually infiltrating the bedrock. It was inadvertently recharging the groundwater, slowly returning water back into the aquifer over months.
And the second service?
The evaporation. The engineers viewed evaporation as a total loss of the resource. But evaporation isn't destroyed water. It is just water entering the atmosphere. All that standing water in the earthen ditches was humidifying the local microclimate over millions of acres of farmland. That intense local humidity was a critical trigger for generating summer rainstorms.
Oh, wow.
Yeah, the inefficient irrigation was physically creating the rain that the farmers relied on. By sealing the entire system inside PVC pipes and highly efficient precision sprinklers, we stopped the aquifer recharge completely and we dried out the local atmosphere, causing devastating localized droughts.
We hyper-optimized for the immediate yield of the crop, but we accidentally broke the entire regional water cycle. We turned a circular system into a linear one. So Enfield proposes what he calls the Ditch 2.0 hybrid model. We don't abandon the center pivots. We need the crop yields to feed the world. But we deliberately bring back managed systemic seepage. We dedicate a percentage of the land to rebuild those biological treatment corridors.
Right, integrate it all.
We fill them with the duckweed to catch the nitrogen, we let the water intentionally seep to recharge the groundwater, and we let it evaporate to humidify the air and seed the rain clouds. We build the inefficiency back in because the inefficiency was actually the planetary infrastructure keeping the whole system alive.
If we step back and tie this entire massive deep dive together, we see that Enfield's architecture is totally self-reinforcing from the atomic scale to the planetary scale. It's a closed, infinite loop of civilization. Let's trace it. The shallow algae ponds that are scrubbing the toxic exhaust of the coal plant, those algae are generating massive amounts of lipid oil for fuel, yes, but the leftover algal biomass is pure, high-quality protein.
And that raw, sustainable protein feedstock from the algae is the exact liquid medium that is pumped into the Optogenetic lithography 3D printers.
Exactly. The macro waste feeds the micromanufacturing. And then lasers are fired into that exact protein vat to grow those indestructible, self-healing amyloid composite materials.
And those amyloid materials are used to build the physical structural infrastructure of the new bio-CCS plants, the biological quaternary computers that run the logistics, and the piezoelectric biosensors that monitor the duckweed channels.
The macro ecosystem feeds the micro-nano manufacturing, and the micro-nano manufacturing builds the tools to manage the macro ecosystem.
Yeah.
The sewage, the carbon emissions, the agricultural runoff, they aren't waste anymore. They are the fundamental vital fuel for the entire technological platform. Biology is the universal unifying technology.
It is an awe-inspiring journey. If you're hearing this, let's just recap the sheer mind-bending scale of what we've covered today. We zoomed all the way into the DNA double helix, where we are taking stripped-down viral capsids, loading them with CRISPR molecular scalpels, and using them as microscopic couriers to debug our genetic code and literally cure diseases like single cell from the inside out. We looked at how we can synthesize the radiation resilience of tardigrades and the limb regeneration of axolotls to functionally translate nature's open-source code and augment our own biology.
It's been a ride.
We explored how the horrifying mechanism behind prion diseases can be harnessed to brew incredibly tough, self-healing piezoelectric amyloid fibers that conduct electricity through tyrosine stepping stones. We learned how to 3D print those exact proteins into computers and medical scaffolds using holograms of light. And finally, we zoomed all the way out to the atmosphere, seeing how duckweed and algae loops can eat our industrial pollution, recycle our fertilizers, and save our collapsing aquifers.
The single most important takeaway from all of this is that the timeline has already begun. This is not distant 22nd century science fiction. Things like systemic in vivo CRISPR editing, Casgevy, and industrial bio-CCS algae plants capturing carbon to make fuel. These are operational online right now. The slow, painful transition from a linear, extraction-based industrial civilization to a circular, biological, programmable civilization is currently underway.
I want you to look at the world differently today when you step outside. Look at a simple houseplant on your desk, or a stagnant pond in a park, or even hold up your own hands and look at them. I want you to realize you are not just looking at nature. You are looking at the most advanced, ancient, and powerful computational and manufacturing substrate in the known universe. We're finally just learning how to type on the keyboard. But I want to leave you with one final provocative thought to mull over. Something we haven't explicitly answered today, but that Enfield's architecture naturally demands we ask.
And what is that?
If we follow this unified biological architecture to its absolute logical conclusion, if we eventually build our cities, our computers, our transportation infrastructure, and our own biological upgrades using self-replicating, light-guided proteins and genetically programmed ecosystems, at what point does our human technology become completely indistinguishable from a new synthetic ecosystem?
That's the real question.
And if our infrastructure is fundamentally alive, if the structural walls of our homes can bleed and heal themselves, if our computers process data using living DNA strands, and if our power grids are essentially vast engineered forests, what rights, what ethical standing, or what entirely unforeseen evolutionary paths might that living infrastructure eventually command? When we build our machines entirely out of life, do we owe our machines the same respect and stewardship that we owe to a virgin redwood forest?
It brings us right back to those diagnostic muddy waters we started with. The rigid binary lines we've drawn between the natural and the artificial, between the patient and the program, between the city and the forest, they entirely dissolve.
They do. The mechanical linear X-ray machine is broken forever. But the true biological source code is finally starting to make sense. Thank you so much for joining us on this deep dive. Stay insanely curious, and we'll catch you next time.